a whole different level.
Yeah.
Okay, I'm going to go ahead and turn off the waiting room just temporarily so we don't have to manually do this every time.
Right.
And then I'll turn it back on when we get close to when it's start time.
Okay. I'm going to suggest that I paste the programming to chat.
That's a good idea.
Then at least we can all refer back to it then, can't we? Do that. Where am I?
You see my fenestraria in flower?
Yes.
Cool.
Very nice.
It's all working then.
Yeah. Oh, yeah.
It's the yellow flowered one, so that's the right rhodophylla.
Or Antioco.
Yeah, Rhodophylla aurantiaca with the yellow flower.
Yeah.
Excellent. Thank you.
I think the Rhodophylla subspecies Rhodophylla is supposed to have a white flower.
Well, I've obviously joined the correct meeting then.
Yes.
I think so.
Yeah, the other one is supposed to be white flower, but there's a lot of thought that it's merely the same plant with two different flower colours.
Mm.
So, Ivor, I've copied your agenda as well, and I'll paste it in between each speaker so you don't have to think about that.
Okay, yeah. Mm.
Afternoon, Chris.
Hi, Alina.
Oh, you're in on two devices, yeah. You've muted your only microphone, Chris.
I'll do that too in a minute here. I'll log in on my iPad too, just in case anything happens. I'll make myself a co-host on that, and so you'll see an iPad or maybe another in my name.
We'll be able to hear you now.
Hello.
Good afternoon, Chris.
Hello. Oh, I'm there. Wait a minute. Hang on. I was looking for the. Oh, that's better.
And Ivor, we did want to record this, right? That was what we decided the other day.
Yeah.
So we're gonna.
Yeah.
Okay.
Yeah, both the major speakers have agreed to that. Yeah, without.
So what about the short speakers? Okay.
Yeah.
So I'll make 3 recordings: one of the main thing, one of the short speakers, and one of the last, or the first speaker, the short speakers, and the last speaker.
Yeah, that's probably a better way to do it. Ian, I can't hear you. I can see a plant being stuffed up my nose, though.
Hello.
Good afternoon. What are you showing me? Oh, you've muted again. You muted again.
That's just me.
No, you're still muted. I don't.
Put your finger on the space bar and hold it down for a second. That's it.
All right. In case you've never seen a Hypanthodium, it's a Dorstenia foetida, and there's its floral structure.
Ah.
Can you see it?
Just about. It's focusing on your face and your background rather than the plant, but yeah. No, it's too far away. Yes.
I've seen that on many a Dorstenia.
Yeah. Mm.
Yeah. Anyway, I just thought I'd show you that.
Thank you.
Nothing to do with Mesembs.
Yeah. John, are you making the speakers for the short talks co-host now or after the first talk?
I'll probably wait till after, like right after the first talk when you bump on, because the biggest cause of interruptions is somebody who can unmute themselves, the co-host unmuting themselves and forgetting to mute again. So
you'll just need to have Terry on because he's
already a co-host. He's already
Ivor, in my bits at the end, do you think we should mention possible future plans?
I don't see why not.
Right, I've got it noted down.
Yeah. Yeah.
Well, one thing I was going to ask for in the introduction was to ask for feedback from people to see what they thought of this afternoon.
Yeah, that'd be good. Yeah. Mm-hmm.
As I keep saying, it's going to be a good meeting.
I reckon, Terry, what we really need for that is the sort of the voting thing that you can put up.
Yeah, which certainly I've never used, and certainly in current setup at the moment. But we need ideas, really.
John might know how to use it. Do you, John?
What's
putting
that?
Putting a voting thing up on the screen. Do you know how to do that?
Oh, we could do a poll, but it's easier to set it up ahead of time. I didn't set it up ahead of time. We could do it if you really need to, but you should probably type what you want to vote on and send it to me, and I'll work on it while he's talking.
I think the other thing, Terry, is if people just want to make comments in the chat, because we're going to record those, then at least we'll have we'll be able to see them afterwards as well, what people have said.
We'll go that way, John. People put things in the chat.
Is my squeaky chair causing a problem for people?
No.
No, good.
As long as it's your chair.
Hmm?
As long as it's your chair that's squeaking over.
It's the chair. It just squeaks. It's old, like its owner. Oh, people are starting to come in. It's good.
Yeah.
Quite a few names I don't recognise.
You see Alex Martin's there?
He is indeed. That's the first time I've seen him at one of these meetings.
No, he's been to a few.
Has he?
I'm sure he can hear you.
Good afternoon, Matt. That's always a good start when the speaker joins.
All right. Oh, good morning for me.
Good morning. Sorry.
Yeah. I changed my name, if I can figure that out.
Hello, Matt. It's probably the first time that we've ever encountered each other. This is Terry here.
Oh, hi, Terry. Yeah. First time.
And I'll be introducing you this afternoon.
Okay, sounds good.
Have you ever been over to the UK?
No, I haven't. Yeah, I've never been to Europe at all. I've been to South Africa a number of times.
Oh, I'm aware of that. Certainly.
Okay, I'm going to go ahead and turn off the bell because we've got so many people coming in now. No, I think we know that people are coming.
G from NL.
Oh, let's turn it on.
NL.
Oh, afternoon, David.
Hello, Ivor.
Yeah, good afternoon, David.
Peter. It'll be a change, won't it? Something different.
Hope so.
So, David, how are you doing with counting all those conophytum seeds?
They're all done, Terry.
Oh, excellent.
And
you've still got your eyesight.
Well, I'm still cross-eyed, but yeah. Silicodon singularis is not the biggest of seeds either, is it?
No, it's not. It's quite difficult to clean up from debris.
I'm sure it is. You must spend hours cleaning the seed.
Yeah, but I'm practiced from the MSG days.
Yeah, but it still takes time, doesn't it?
Oh, yeah, yeah.
Mm.
I think finding them's a difficult bit.
Finding them?
Yeah.
You hold the packet upside down and flick it and it goes into the pot. You might not see the seeds, but they are there.
Afternoon, Terry.
Good afternoon.
Chris Leather seems to have appeared twice. Once as a sunflower.
As the afternoon goes, afternoon, Terry. Ian, as the afternoon goes on, you may well be grateful for Chris appearing twice. He may well have resolved a problem by his double appearance.
The mystery thickens.
The mystery thickens, yes. When we did a test yesterday, our other main speaker was unable to access the microphone on her computer. So we had to use the Chris Leather double take to get her voice into the meeting.
Yeah. That sounds like a familiar problem, doesn't it?
Yes, thanks for the idea, Chris. It worked out well yesterday.
Afternoon, Andy.
I think I see a few people.
Andy's muted. Can you unmute Andy?
Oh, I'm here.
If you want to talk, it's up to you.
Oh, I'm not sure. We'll see.
Good afternoon.
And you?
Thank you. Has Chris appeared yet? I've not seen him.
No, I was emailing him just a couple of minutes ago. He is around.
Yeah. We should be all right. 46 at the moment, so not too bad. Just awaiting everybody else to join us.
Yeah, I think the BCSS finds that everyone turns up in the last couple of minutes.
Yes. Yeah, everybody's turned up on the last second, yeah.
Just another 10 minutes.
Yeah, time yet.
Yeah, they'll turn up. They'll turn up.
Yes.
No, I didn't get mine.
What's that? Oh, the journal arrived today. Arrived today.
Yep.
Here it is.
Excellent. Somebody complained that the postman had dropped theirs in a puddle. Could they have a replacement?
I can prove it.
Doesn't get what to go and do.
Suzanne, I got my Bacchus this morning.
You did?
Yeah, and it's not Melissop, as other people think it might be.
No, quite. Have you seen that there's another one gone up?
Ah, but it's not.
It's not, okay.
There's one gone up as a Bacchus, and it's not a Bacchus.
Right. Okay.
This is probably totally cryptic to everyone else there.
Yes.
It is intriguing.
And it's not wine either. Something for you to work out what Terry and I have got in common, apart from Mesembs.
Paperweights.
Yep.
Now, how did you guess that?
I wonder. I too have been in your lounge.
The email address is a giveaway.
Yes, indeed.
Just a bit, yes. Something else went quite well for you this morning, Terry.
It did indeed.
Yeah.
Andrew, I saw you have your MSG pin. I want to go get my pin now. On your shirt.
Yeah, I put mine on today.
Ah, very nice.
Don't forget, if anybody hasn't got one, we've got plenty in stock.
Oh, that's so.
Oh, but they're all reproductions.
Oh, go on.
They're not originals.
They're not facsimiles.
Are we going to get a Northwest Mesemb show on, Peter?
Got one.
I haven't thought about it.
Just waiting for the MSG paperweight, surely.
Mesemb.
Hi, Andy.
MSG show.
Hi, Matt.
Usually held in Manchester. Used to be in Alan Hart's Hartford.
Oh, yeah.
Hi, John. Played any cribbage lately?
Afternoon, Derek.
Hello there, Ivor.
You well?
Yes, except I've been cleaning greenhouses this morning, washing them down. Hello.
It's good for you. Mine's had a good wash this morning on the outside.
So, were you doing it in the rain?
Yeah, it's pouring down.
Well, that's the best time, I would say. You don't have
Yeah.
to.
Well, I didn't do anything. I just stood back and let nature take its course. So far, we only seem to have two of our speakers for the middle pet section, don't we? A couple more missing still.
We've got 6 minutes left, Jack.
Yeah.
I've got an hour and 6 minutes, thankfully.
They're all putting their makeup on.
Yeah.
Oh, numbers are going up.
Oh, Alice has just arrived.
Oh, good.
It's a bit depressing news, I'm afraid, but I've just had an email from Peter van Wyk, who's the one of the rangers up in the Richtersveld National Park. And they just announced some proposals to fully develop the Alexander Bay area, to build a oil terminus and a deep water port, build residential and light industry developments on the lichen fields, and a railway line from Alexander Bay all the way down to Cape Town. It's quite incredible.
And the lichen
so if anybody's interested, just get in touch and I'll send you what some of the details are. It's not happening yet. I think it's proposals to seek funding, presumably from
those lichen fields are most unique habitats.
Yeah,
I know there are one or two others in other places of the world in similar areas where you've got fog coming in off cold currents, but it's a very, very restricted vegetation type.
Well, Norbert Jürgens, the German academic, he's replied already, and to express dismay, made exactly the same point. I've seen the lichen fields up in the Skeleton Coast, and they're nowhere near as dense as those in the Alexander Bay area. They're just pristine. They're fantastic. But if anybody wants details, I can forward just send a chat to me and and your email address if I don't have it already, and I'll send you what information I've got. If you want to be depressed.
Well, I'd like the information, but I can't send you a personal chat because only the co-hosts are chattable.
Oh,
apparently.
Let me work on that.
Anyway, Andy, send me the information, please.
Who said that? Sorry, well, Al. Okay, yeah. See you now. Yep.
Yeah. That's a red jumper.
Oh,
You're there. Yeah, at the top.
We're nearly up to 100.
Yeah, 99.
So on my iPad, which isn't logged in as a co-host, oh, it is. On my iPad, I can see the ability to chat everybody. Let me test it again.
In the morning.
Yeah, so we didn't use the Manchester branch site. We've now exceeded 100.
Yep, 101 now.
Oh, that's good.
And
Yeah. Good afternoon, everybody. It's good to see some very familiar faces from days of old and more modern.
Hello, Roland.
Afternoon, Roland. I was trying to see who it was speaking then. Didn't quite recognise the voice. She was a bit too far from the microphone.
Oh. I said hello, but it was muted.
Hi, Ian.
Yes, she's there.
Hello, Terry and Jennifer. Jennifer peeping round.
Well, it's now 1 minute to 2, and as we've got to keep strictly to time, I'm going to ask John if you should mute everyone, or shall I mute everybody? I'll mute everybody. That'll be easier. So I think everybody should be now muted. Oh.
No.
Oops.
No. 1, 2, 3 at the moment.
Yeah. Oh, not everybody is muted.
No.
No.
Try now. They are now. Terry, I think you can unmute yourself.
I think I have done so.
Excellent.
Yes.
You've got about 30 seconds, and then we can be off.
Okay.
When the clock ticks to 2, we can let you go. I shall quickly say thank you for everybody for attending, for our experiment at trying to get a convention through Zoom. I hope it works well. Thank you to all our speakers who volunteered to take part and everyone who's turned up. And I shall hand over to Terry to introduce the day.
Okay. Thank you very much. Well, good afternoon, everyone. Or even good morning to our friends in California, or good evening to anyone in Asia and Australia. I know one of my friends in Adelaide was intending to join. Now, for all of us, it's been a very unusual year. We haven't been able to get together at all. And before March or April, I don't think any of us had ever heard of Zoom. And now I think most of us wish we'd actually had shares in the company back in March. So many things have now moved into the sort of online Zoom platform. And this is actually something that we can take advantage of. This will be the first time, for instance, that the MSG has ever had the chance to speak to most of its members all at the same time. So anyway, Ivor Crook, who's our host here, he suggested that we replace 2 live Mesemb events, which should have been last Saturday and next Saturday, with this one online event. And Manchester branch that Ivor belongs to does have a 100-person Zoom license, but John Schwark in San Francisco, the San Francisco Cactus and Succulent Society, he offered to let us use their license for the afternoon, and we're very, very grateful for John for doing this. Now, the 2 meetings that this is replacing, first of all, the Mesemb study group, which was established by Suzanne Mace in 1986 with the help of 1 or 2 other people. That soon started doing a biennial event, usually in the London area, but we haven't actually ventured further away from London than Reading in all these years. But every 2 years we've had this event with plant displays, plant sales, auction, and a talk from a suitable visitor. Up in the north, the Mid-Cheshire Mesemb show was established around at the same time as the MSG back in the 1980s. And they have an annual event, which is very similar to the MSG one, except it's competitive. And the show part is competitive, which ours isn't. But, you know, these are the two events that this is replacing. And I think what we'll miss really is the social aspect, because, you know, a lot of us get together at these events and just chat with each other. And also the plant sales, because what you can buy from nurseries is actually fairly limited. But what's produced by amateurs and sold at events such as ours really helps people to build up their collections. So these are the two things that we're going to miss from this. And I hope that as soon as possible, we can also, you know, we can get back to some live events. But at the same time, you know, we'd very much like feedback at some point about the event this afternoon, because, you know, maybe you'd like the MSG to, you know, maybe put on occasional talks for the study group. So please give us some feedback on that and, you know, possible things that you would like us to do. I suspect one or two people are joining us now from other countries. In the Mesemb event at Banstead, we usually get people from overseas. We've had people from the USA, Australia, China, Korea, and most of the European countries. And I hope that at least some of those are joining us virtually this afternoon. Now, this afternoon's programme, we've got talks from two main speakers. Matt Opel will be the first, and I'll introduce him in a minute. And at the end of the afternoon, Robin Powell, who is a South African botanist currently working at Kew, will be giving a presentation, which will be introduced by Peter Bint. And in between, the sort of member sandwich, when 5 Brits, one of them actually being an expat Brit living in Spain, will give us some short presentations about their collections. And it's going to be a long afternoon. So there'll be 2 short breaks, hopefully about 10-minute breaks if we don't overrun too much in the course of the afternoon. So you can have a walk around, visit the loo, have a beer, have a cup of tea, whatever it is you fancy. And before I introduce the first speaker, just remind you of something that you can do. When we're into the screen share mode, I'm looking at the presentation, you can get rid of these video panels at the side, showing the likes of people having their lunch. If you go to the top of the screen where it says View Options, click on View Options, and then click on Hide Video Panel, which appears as one of the options, and then you can get rid of the images of people while you're watching the talk, and they can come back again automatically later. So now it's my pleasure to introduce a person that I know by repute very well, but who I've never actually met, and that is Dr. Matthew Opel, who is a botanist and horticulturist at the University of Connecticut, where he works for the Department of Ecology and Evolutionary Biology. Now, Matt, as such, is one of these rare creatures, and that he is both a botanist and horticulturist. Very often people are one or the other. So not only does he study them, but he grows them as well. And I know that he grows them very well. Now, much of Matt's work has been on conophytons, and he's done papers on things like evolution and morphology, and particularly looked at microscopic aspects, such as things like seed structure and leaf anatomy and flower pigment patterns. And then he's helped to relate these to the classification of the genus. And you probably best know him for the dumpling, where he contributed a couple of chapters on leaf structure and chromosome numbers. So with that, he worked very closely with Steve Hammer. And in more recent time, he's worked closely with Andy Young and Chris Rogerson. So very much he's been collaborating with various Brits as well. Now, I know that Matt has done a lot of field work in South Africa, and I think he must have a very good eye, because as an example, Jennifer and I tried to find a conophytum at its only known locality until Matt started working. And even where we knew precisely where it grew to a couple of meters, we couldn't find it. Yet Matt goes and finds this elsewhere, you know, growing in a completely unknown place quite easily. And he seems to be able to identify small plants because, in particular, he discovered a very tiny Tylocodon, which is named for him, Tylocodon opal EE. And it's actually a very nice plant. It's easy to raise from seed, starts flowering at a couple of years old. It's a really lovely tiny miniature. And I know he's interested in things like pelargoniums and certainly interested in aerospermums. And he uses his plant collection to produce seed. And some of that seed has been kindly donated to the MSG over the years. And if you're growing anything under an MRO number, that's originated from Matt. So thank you very much for agreeing to talk to us this afternoon, Matt. And your talk is on winter mesembs, cultivation and taxonomy of selected genera. So over to you.
Well, thank you very much for that extremely kind introduction, Terry. And thank you all for having me out here and for showing up this morning or this evening or whatever it is for you. Let me see if I can get the screen sharing going here. All right. Okay, so my talk is going to be on winter growing mesembs and covering Conophytum, which is sort of my area of specialization, and a lot of other things as well. Yeah, let's get started. So the mesembs, I'm not sure how much I need to go into this stuff for this crowd, but they're members of the family Isoaceae now, and with a certain specific set of characteristics. The fruits are hygrocastic capsules, which means they open up in response to moisture in the environment. The flowers are not thought to have true petals. Instead, the petal-like structures are modified stamens called petaloid staminodes. And the epidermis, at least primitively in the mesembs, was covered with bladder cells, although many groups have evolved different sorts of epidermis in the course of their evolution. So it's the former family Mesembrianthemaceae, but the two subfamilies in the Mesembrianthemaceae, the Mesembrianthamoideae and the Ruchioideae, have now been included in the family Isoaceae. So it's a fairly large family, about 1,700 species, by far most diverse in South Africa. So most of those species are in South Africa, and it turns out that most of those species in the Isoaceae are actually in the former Mesembrianthemaceae, what we sort of informally recognize now as the mesembs. So it's maybe about 100 species in the other couple of subfamilies that are now included in the Isoaceae. So most of that diversity in the family is mesembs, and most of them are Southern African, and particularly in the Republic of South Africa is where they're most diverse. They're all leaf succulents, at least to some extent, with other succulent parts. and commonly known as the ice plants because of the bladder cells, which kind of sparkle in the sunlight like ice, or midday flowers, or the feihies. I think I'm pronouncing that more or less correctly. My Afrikaans is a little bit rusty. So here's a map of their distribution. And like I said, yeah, you can probably see my pointer. Almost all of them are native to Southern Africa. So they come from various parts of the world, but say out in Oceania here in Australia and New Zealand, there's not that many species, and it's a lot of things like Tetragonia, New Zealand spinach, which is not one of the Mesembs. It's one of those other subfamilies in the family. So most of the plants that we're interested in cultivating in our greenhouses and on our windowsills and under lights in the basement are South African plants. Most of the actual Mesembs come from that area as well, although a few get up into other parts of the world. There's some Delospermos in East Africa and Madagascar, and some Carpobrotus and Mesembrianthemums are quite widespread, especially in coastal regions. So some of the characteristics of the Mesemms, those two subfamilies, the hygrocastic capsules here, it's thought to be an evolutionary adaptation to protecting the seeds during dry periods and then only exposing the seeds and dispersing them when moisture is available for germination. So during rainstorms, the capsules, which would otherwise remain closed after they're dry and mature, they open up and the seeds are exposed and can be splashed around and spread around the habitat. So it's a sort of a protective mechanism for the seeds. And a characteristic of those two subfamilies. And just a few species in those two subfamilies have lost that characteristic, like Carpobrotus has fleshy fruits that are edible and presumably dispersed by animals. So they don't have hygrocastic capsules, but the ancestral state in the Mesembs is having these capsules that open up in response to moisture. Bladder cells, so in the epidermis of a lot of Mesembs, there's these water-containing cells that kind of sparkle in the sunlight and give them the name ice plants. And again, that's probably the ancestral condition in these subfamilies. Quite a few species have lost this characteristic. So some of the ones, especially for more arid regions, have a xeromorphic epidermis, so sort of a hard, tough epidermis. They've lost these bladder cells, and they have sort of a more drought-adapted sort of epidermis. All right, so the winter growing part of my talk title. This map shows rainfall patterns in southern Africa, and the sort of bluer areas here, dark blue or light blue, are the parts of this area that receive most of their rainfall in the winter time. So it's kind of centered around Cape Town down in here, and that part of southern Africa has more of a Mediterranean climate. The further you go north into Namaqualand and into the Namib Desert, the more arid it is. And also out to the east in the Little Karoo and the Tankwa Karoo, it gets more arid as well. So it's more of a winter rainfall desert type environment. And that's where a lot of the plants that we're interested in come from. So the basic structure of the talk, now that the introductory part is out of the way, is going to be, I'm going to talk about cultivation for a while. And so yeah, I'll talk about how I grow these plants and have some other people grow these plants, some techniques you can use, and what they need in terms of light and water and whatnot. And then I'll do sort of a whirlwind tour of winter growing Mesemb diversity, and cover various groups and sort of arranged with you know, how they're behaving throughout the year. So probably the most important aspect of cultivating these plants is the light that they get. And in general, for most of us in more temperate climates, the general rule is as much light as you can get them. It's probably not going to be quite enough for their winter growing period. During the summertime, these plants will often benefit from some shade if they're in a greenhouse, or you know, a little bit of reduction in their light levels so they don't burn. They often can you know, suffer from excessive heat or sunlight in the summertime when they're dormant, and you can lose plants at that time of year from burning. But in the winter time, probably as much light as they can get, is what you want to give them. And I do grow some of these things just in windowsills. So here's one of my pet conophytums that just sits in the bedroom windowsill. And it's a south-facing window, so for us in the northern hemisphere, that's where they want to be. So they get good sunlight. And it's enough for the plant to do okay, but it's, you know, this Conophytum obcordellum here is a little bit stretchy. It's not really the way I'd want to see it ideally. But it's, you know, it's healthy enough. It does flower and sometimes sets seed if I pollinate it. But really, this one up here in the inset in the corner, that's a plant out in the greenhouse, and it's in a lot better shape. It's a lot more compact. It's got better coloration of the leaves. So a greenhouse sort of situation, a good sunny greenhouse is ideal with really exposure to full sun and no shade during the winter months, or in the fall and the winter up into early spring. And then if it's in the greenhouse, some shade in the summertime to prevent burning. I do grow plants at home under lights, and you know, with good supplementary light, you know, maybe in a windowsill, but to provide some natural light, but with strong artificial light. It's possible to grow the plants pretty well, I think, indoors without a greenhouse in a home situation. And some of the people in the local cactus clubs who have greenhouses are sort of wishing at this point that they had, you know, stuck with a basement light setup rather than trying to grow them in greenhouses, because it's, you know, it's easier to control the light levels, to control the temperature in the environment, and it's cheaper on the heating bills as well in a kind of sort of cold temperate climate like we have here in Connecticut, at least. So I used to have plants under fluorescent lights, and these are daylight sort of light bulbs that are used in the fluorescent lights. 6500 Kelvin is the color range that you want. So sort of a harsh blue daylight type bulb is what you need. Fluorescent lights are probably a little bit on the way out, and I've been switching over to LED lights, the new technology coming up. And so here we've got some of my current setup, which has got, you can see in the center here, some specifically made LED grow lights. These are wide spectrum ones that still have a little bit of that purple evil glow, which is a little bit hard to look at. It's making my head hurt even just looking at this slide. And I have a couple of, as well as the LED grow lights, I have a couple of LED shop lights, you know, just sort of more regular consumer lights, but again, sort of a daylight spectrum on those. And that provides a little bit of extra light and maybe cuts down a bit on the purple color and makes the plants look a little bit more natural when I'm trying to work with them under the lights. And these plants are in a windowsill, and these LED lights are so bright you can't really tell, but outside it's actually the middle of the day, but it's hard to tell because the photograph is you know, so overexposed to these very bright lights. So that's the current setup. These LED lights are probably about 40 watts of actual power consumption. And the labeling is always weird. I think it says 100-watt bulb on the package, but it's the power consumption is actually 30 to 40 watts. So it's a couple of those set up maybe a foot or 30 centimeters from where the plants are, and arranged so they're kind of evenly illuminated underneath. And that's given me good results. I have experimented a bit with the higher wattage fixtures, probably intended for grow rooms for cannabis. And they're often a little bit too much for growing plants on shelves and growing small plants close to the lights. It's possible to get scorching and sunburn with LED lights as well if you've got a little bit too much power. So you've got to kind of experiment with the wattage you're putting on your plants and how close the bulbs are and what your ventilation is like. So temperatures. Which is an especially cold spot in South Africa, but a lot of the places where our plants grow do experience frost on probably a more or less annual basis. Snow might be a little bit more rare, except for up in the higher mountains. But a lot of the plants that grow, the conophytums and whatnot, are used to quite cool temperatures in the winter time, and we wanted to, at least some extent, try to emulate that when we're growing them at home. So probably about 10 degrees Celsius at night, and getting up to more room temperature, more comfortable temperatures during the daytime is probably kind of ideal temperatures for growing these plants. They can experience colder temperatures in the wild, but it's not necessarily ideal for them. So the plants might experience in their native habitat, but it is different what the plants can tolerate versus what is ideal for them. And especially what they can tolerate in a pot might be different from what they can tolerate if they're growing out in the ground and have that sort of thermal mass of the soil all around them to moderate the temperatures a bit. So I don't recommend exposing the plants to frost, although probably over the years, a certain number of my plants have been forgotten outside a little bit too late and experienced a bit of frost, and I haven't seen a whole lot of damage from that, but best to keep them above freezing at least. But they do appreciate that strong cooling off period at night during the fall and the winter and the spring when they're most active. During the summertime, you know, in habitat, a lot of them probably experience temperatures, quite hot temperatures up to 40 C or something like that. Again, it's probably best in a pot to protect them from those sorts of temperature extremes. You'll also notice in this slide that things are quite wet sometimes in the wintertime where these plants grow. And you do see in habitat, you might see plants growing in areas that are even flooded a little bit, little pans in the rocks that flood during the rains, and you'll actually see conophytums and crassulas growing underwater, at least briefly. And again, that's the sort of thing that you might not want to try and emulate in a pot, but it shows that the plants can, at least in habitat, tolerate a fair amount of moisture during their winter growing periods. So you want to water them pretty regularly during the winter time. You don't want the plants to sit around wet, but you don't want to get them to the point where they're sort of totally dry throughout the entire volume of their soil mass in the pot. So having a little bit of moisture in the soil, at least in the lower layers during their growing period, is probably a good thing. And exactly how often you have to water them depends strongly on your conditions and what the humidity and the temperature is where you're growing them, and it's going to be very variable from person to person. But for me, under the lights, I probably water my plants about once a week or so this time of year, and let the surface layers of the soil get fairly dry in between waterings, but try not to let them dry out completely. And you've got to watch how that's at this time of year for us in the north would be really starting to grow right now. If they're still wrinkly or still very dormant, you probably want to up your watering a little bit. You can see the leaves crack open. If they're getting too much water, that's a sign that you've got to cut back a bit. Soils always a controversial topic, and even for myself, I probably go back and forth on what exactly I want to include in the soil and what my recipe is. It's experiment with new things, and sometimes they work out well and sometimes not. It's probably not critical exactly what the components of the soil are, so long as it's kind of a neutral pH or a little bit acid, but quite loose and well drained. and not too many nutrients in it, not too overly fertile, which can also cause the plants to urge and start to crack open, and you can get problems with fungal diseases and whatnot, and rotting and those kinds of conditions. So sort of a basic soil that I've used over the years, and this is kind of the basic soil that I use. And depending on where you are, even certainly for an international audience, exactly what soil components are available is going to vary quite a bit. So you may not have all these things available to you, or you may have probably some more or less equivalent substitutes which you can use for your soil mixture. So for especially the conophytums and the more compact Mesemms, I tend to base the soil on sandy loam soil, and so a native soil mix. And sandy loam in the sort of soil science sense, so a soil that contains some sand, but also more silt-sized particles, kind of particles in between clay particles. So a little sandy loam subsoil here without much organic material. 1 part of that, 2 parts of sharp sand. You want to avoid things like beach sand or very fine sand, which can kind of clog up the pot and cause poor drainage. 2 parts of vermiculite and 2 parts of perlite, which are both expanded volcanic rock products, which are light, hold a little bit of water and nutrients, but not too much, and provide some good aeration and drainage. And then another part of coarser mineral material. This is turfus, which is a calcined clay product. So clay fired at a very high temperature, so it's won't dissolve when it gets wet. It's fairly stable. You can substitute in, instead of this, pumice. People on the west coast of the US use quite a bit as a good amendment, or sort of small pea-sized gravel is a good amendment. I sometimes base my soils with a soilless mix, a peat-based or a coir-based mix. It definitely can work out. Some very excellent growers that has some organic components there, soilless mix, and they do well with it. You just gotta, again, watch your plants and adjust your watering and fertilizing sort of in accord to what you have. For fertilizer, by the way, with adult plants, I do very little. I'd say maybe a couple of times during the growing season, I give them a light watering with a typical water-soluble houseplant fertilizer, but diluted to about half strength or a quarter strength of what it recommends in the label. So you don't want to fertilize them too much, and you want to watch how the plants are responding to it and make sure they're not getting too green and stretchy, which they might do if they get too much in the way of nutrients. A little bit of propagation. Cuttings in winter growing Mesemms are probably best done in the fall, at the start of the growing season. And I should say at this point, people always ask, there's usually somebody who asks, these are mostly southern hemisphere plants, so if you got to keep that into consideration, if you got to take that into consideration and water them in April, which is the southern fall, is that when they want to start growing? And that's definitely overthinking it. When I say that these are winter growing plants from the western parts of South Africa and southern Namibia, they grow when they start growing when the nights are cooling off and the temperatures are getting lower, when the day length is getting shorter and the nights are getting longer. That's what they're responding to. So a plant of conophytum that's growing in Worcester, Massachusetts, doesn't know that it's the 6 months reversed in the opposite season in Worcester, South Africa. So the plants are responding to the local conditions. They have no idea what's going on in South Africa. And even imported plants adjust quite quickly to the season and the part of the world that they're growing. So when I talk about starting cuttings in the autumn, that means wherever you are, the season where the days are getting shorter and the nights are getting colder, is the best time of year for starting the cuttings, and you shouldn't necessarily worry about what's going on in South Africa if you're not in South Africa. So the cooler part of the year in autumn is probably the time of year to start cuttings, and they're generally fairly easy for a lot of winter mesembs. You can use rooting hormone preparations, but they're probably not necessary. Most of these you can cut off a few heads, a few pieces of stem, and probably in a month or so start to get rooting if it's the right time of year. You can start cuttings at other times of the year, but often if you start them, say, in spring or in summer, they'll just sit for months without doing anything, and they may root just fine in autumn, but it's kind of taking a risk of the plants drying out completely and not getting a chance to grow before they dehydrate and you lose the cutting. One other thing about cuttings in mesembs is that, with some succulents, you can propagate them from leaf cuttings. As far as I've known, and I'd like to hear otherwise if anybody knows, mesems can't be propagated at all from leaf cuttings. So you've got to have a little bit of stem involved and an apical meristem at the top of it in order to actually regenerate a new plant. So I have had cuttings of conophytums or lithops that just contain leaf tissue that do produce a little bit of root, but they don't seem to have any ability to regenerate a new stem from that, and the little bit of leaf tissue that is rooted will just die eventually if it doesn't have a bit of stem there in order to continue its growth. Propagation from seeds, and this is a shot of Steve Hammer's seed propagation area at the end of the winter, and some mesems in there and a few other winter growing things. In order to obtain seeds in conophytums, if you want to sort of build up a stock for trading, or for generally have to cross-pollinate two different plants, so almost all mesems are self-incompatible. They're not self-fertile. Can't get seed by just sort of moving pollen back and forth among the flowers of a single plant. You have to have two separate seed-grown, genetically distinct individuals to cross-pollinate in order to get seeds. And people use various things for transferring the pollen, like paintbrushes and whatnot. I've found often that bits of dried plant debris, like this is, I think it's an inflorescence from a crassula here, little bits of grass or crassula inflorescence or pelargonium leaf bases provide good tools to move pollen back and forth. You just have to choose something that's the right size to get into the business part of the flower. Some conophytum seedlings here. The seed of most mesems is quite small, so sort of dust-like to maybe at the outside the size of poppy seeds or so. So most of these have to be sown right on the surface of the soil, and they also benefit from having some exposure to sunlight in their germination. And for germinating the seeds, I like to keep them on the moist side, but pretty much as soon as I start the seed germination, you can start the seeds with a bit of plastic covering your pots or something like that to keep up the moisture levels. But trying to grow on the seeds in an enclosed environment has never really worked out very well for me. Seeds, like with cuttings, I like starting them right at the start of the growing season. So in early autumn or even at the very end of summer is a good time for me, kind of the whole cool season to develop and to put on some mass and to get ready for their first dormancy in the subsequent summertime. Especially, I think, from growers in England and in sort of cooler, more maritime parts of Europe, like the Netherlands, that you can start seeds in the middle of winter, or even in the springtime, and kind of keep the seedlings going over the summer. I have a very hard time doing that. The seedlings really like to go dormant for me in the summertime as soon as it starts to get hot and sunny. And if they haven't had the whole winter season to develop. They're often not big enough to survive that. Seeds other times of the year and it works for you, I'd say go for it. But so starting things in the autumn is what really works for me. Seedlings need a bit more coddling than adult plants, so they need a little bit more moisture and maybe a little bit more regular fertilizing with a quarter strength liquid fertilizer. Every couple of weeks maybe, or even every week when you're getting them started. Again, you want to allow the seedlings to put on enough dormancy, and they start out quite small, so it's good to push them a bit when they're starting out to ensure that you've got a plant that's large enough to survive its first warm dormant period. And a little bit of trouble in paradise with this particular shot of Conophytum seedlings. Most of these are healthy seedlings, but notice the couple down here that look a little bit glassy and translucent, and they're a little bit duller than the seedlings in the other part of the photograph. This is a shot of a pot that's having a little bit of trouble with damping off, which is a disease, especially of seedlings. It's a problem with Mesembs. So this can occur, especially if the seedlings are not quite getting enough light, maybe, or if they're a little bit too moist, or if there's can cause problems with this. It can be a problem. It seems, just based on what pathogens are in the environment. So sometimes even pots that I would think are being given enough light and enough air circulation, you can sometimes get problems with this, especially in some of the more delicate species. Damping off is probably in this case caused by an organism called Pythium, which is, people think of it as a fungus. It's not technically a fungus, but it's sort of for horticulture purposes, you can think of it as a fungus. And it does respond to certain broad spectrum fungicides are labeled for Pythium and damping off. And you might have to make use of fungicides in some cases if you're having persistent problems with damping off. And cultural remedies don't seem to be working for you. So another problem with disease I sometimes see in Mesembs is these kind of light colored spots on the leaves. And it seems to affect certain shrubby Mesembs, especially like Dracophyllus and Undulateria. I sometimes see it on Conophytums and things as well. And it's generally doesn't seem to harm the plants too much, but it's quite unsightly. And I believe that this is a form of white blister, and it may be the genus Albugo, may be the genus of the pathogen. And this, like Pythium, is another oomycete, which is not technically a fungus, but as far as treatments and horticulturally can be sort of considered a fungus. These white blisters do seem to respond to repeated sprayings with a broad spectrum fungicide. And you know, it may be something to look out for in your collections. And if you can, you might want to discard plants that are showing signs of this, or try treating them repeatedly with fungicide. And it does seem to go away eventually if you keep on top of it. I'll say a little bit about pests as well. Mesembs are, for the most part, I've found pretty free of insect pests. I, get little problems with thrips on the flowers sometimes, but it's not really a big deal. I'd say the main insect pests that affects a lot of Mesembs is root mealybugs, and those sometimes can be cleaned up by very carefully unpotting the plants and knocking off all the infected roots and washing them off and keeping on top of them that way. And that can work in a small collection, especially. You might have to move to a systemic insecticide for larger collections to treat these. And root mealybugs do seem to be susceptible to a lot of commonly used systemic insecticides. So You can use insecticides according to the labeled directions and often take care of root mealybug problems, but you want to occasionally unpot your plants and take a look at what's going on. Root mealybugs will appear as very small, kind of waxy or cottony insects on the roots, especially on the edge of the pot is where they tend to like to congregate. So my whirlwind tour of winter Mesemb diversity is going to be sort of based on, I'll talk about some different groups of Mesembs and try to cover a number of different genera and kind of cover a full year in the Mesemb greenhouse and what's going on with the plants at different times of the year. So here's a little collection of conophytums this time of year in the autumn. And they're starting to grow, and a lot of the dwarf Mesembs, lithops and conophytums will flower primarily in the autumn. There are a few species of conophytum that flower at other times of the year, but they're mostly putting on their big display right now, for me in the Northern Hemisphere at least. Lithops. We typically think of lithops as being more warm weather growers, and most of the species are probably from more inland areas than the winter rainfall region in southern Africa. So most of them are from areas that receive kind of spring and autumn rain and maybe a few summertime thunderstorms. But there are a number of species of lithops that grow in the winter rainfall area, right alongside things like conophytums and adromiscus and whatnot. And Lithops marmorata is one of those. And it may be worthwhile looking at your lithops collection and figuring out which ones grow in more winter rainfall regimes. And I found that some of those, Lithops marmorata, Lithops divergens, often the seeds will germinate a bit better if you plant them in the cooler months than they do if you try to plant them in spring, like you might with more inland lithops like Lithops leslii. Merophytum mayeri here, just starting to grow in the autumn. And you can see the pair of old leaves from last year. It went dormant and these leaves formed a sheath around the new buds underneath. And the new leaves are just starting to break out at the beginning of the cool season here. Dactylopsis digitata, the hitchhiker plant, just starting to grow in the autumn. And look at the bladder cells here on the bit of the stem. When these get larger, the leaves get quite large and they kind of hide the stems and you don't see them. But the stems are kind of neat things. They have these very thick layers of bladder cells on them, which you can see the little bit of green stem underneath and this very thick layer of bladder cells on top. And some of the conophytums, again, starting to grow as the nights cool down this time of year. This is Conophytum obcordellum, a form that used to be called picturatum, which is maybe especially showy from the Clanwilliam area in the Western Cape of South Africa. Conophytum bilobum here. It's a very widespread species in the genus. Grows over a large area in Namaqualand, and it's quite variable over that range in terms of the coloration, the shapes of the leaves, and the texture of the leaves. This is an especially large form from kind of the area east of Port Nolloth, which has a lot of fuzz and trichomes on the surface of the leaves. Conophytum regale here, blooming in the fall. And this is quite a nice plant. It's one of the more bilobe types of conophytums, but with large windowed areas in the sides of the leaves. Quite a beautiful thing, especially in flower. And quite rare in habitat, apparently. It grows in a little area north of the town of Springbok in Namaqualand and nowhere else. And apparently not very many of them in the wild. Conophytum minutum here. And this is in this photograph of the flower here, you can see a little bit of a transition between these petaloid staminodes, the big purple petal-like structures, and more filamentous staminodes, so sterile stamens that are on the outer parts of the flower, really modified to look like petals. And you can see a transition in the inner part of the flower to more filamentous structures. And the actual fertile stamens are kind of hidden underneath those filamentous staminodes in this particular species. So there's a little bit of a transition between regular fertile stamens and these modified staminodes, which help to attract pollinators. So these plants in the wintertime, kind of at the height of their vegetative growth, and some of them flower in the middle of the winter as well. So go over some of these. This is just a shot of the greenhouse at the University of Connecticut, showing some winter mesembs like the Caridopsis here, and also some other winter-growing succulents and pelargoniums and dodaleas and things. Dactylopsis. So we saw this in the fall. This is in full growth in the wintertime in habitat. And Dactylopsis is native to the Knersvlakte region in sort of a bit north of Cape Town in South Africa, sort of the southern limits of the really arid type environments in the winter rainfall region in South Africa. And the Knersvlakte is a region, sort of low-lying region, a bit inland from the coast, where there's a lot of areas that are covered by white quartz pebbles. So there's large areas that are covered by quartz pebbles, and that forms a microenvironment which is quite conducive to the growth of dwarf succulents, apparently. So there's a whole lot of plants like Dactylopsis that are native to that environment and endemic to that area. They don't grow anywhere else. Dactylopsis is one of a couple of genera that I'll talk about right now that are in the subfamily Mesembrianthomoideae. So it's one of the two subfamilies that are involved in the mesembs. And Dactylopsis is one of those genera in the Mesembrianthomoideae. There's been a little bit of taxonomic changes, well, actually some fairly major taxonomic changes in the Mesembrianthomoideae in recent years. And there's been a bit of a move to actually collapse all of the genera in the Mesembrianthomoideae into a single genus, Mesembrianthemum. So this is sometimes considered part of the genus Mesembrianthemum. I tend to still break out a lot of the old genera and keep them separate. There's putting things like this and a lot of the other things I'll talk about into the genus Mesembrianthemum, I think maybe hides a little bit too much of the diversity of the genus and kind of varies a lot in that single genus. Dactylopsis in cultivation, they never really look as good in cultivation as they do out in the wild. It's very difficult to get them as large and as well formed as they get out in the wild. In their native habitat, they have these giant sausage-like leaves, and they get to very large size, and they always look great if the rains have been good. And it's really hard to get them to look like that, and even in a sunny, well-ventilated greenhouse. A lot of sun and very cool temperatures seems to help out, as well as fairly generous watering during the wintertime. But it's tough to get them to grow the way they do in the wild. It's a plant in its native habitat where the soil has often got a little bit of salt in it. And people mess around with sort of adding a little bit of table salt to their water sometimes when they're watering Dactylopsis. I wouldn't recommend that. It doesn't really seem to help, and I think it can cause a lot of heart damage to the plants as well. It doesn't really work out as something to do in the pot to add salt to the soil. So I definitely would not recommend that. Don't ask me how I know. This is Dactylopsis littlewoodii, which is sort of the northern form of it. It comes from the northern parts of the Knersvlakte, but sort of a dwarf form of Dactylopsis. Aspisoma is a shrub from northern Namaqualand and kind of the coastal areas, the western parts of northern Namaqualand. And it's actually the closest living relative of Dactylopsis. So it's a another member of the subfamily Mesemb, sometimes included in the same genus as the rest of the members of that subfamily. But I'll break it off here into its own monotypic genus. Philobolus is a fairly large genus. A lot of them are kind of shrubby and maybe not particularly attractive in a pot, but there's a couple of geophytic species with tubers underground, and those are maybe especially attractive pot subjects. They're totally deciduous in the summertime, so very, very strongly winter growing. They die back to just these underground tubers during the warmer months. Scaledium is another subfamily Mesemb genus, and it gets that name because the old leaves form kind of a skeletonized structure. The veins stand out and they look a little bit skeletal. Scaledium, if you try and look it up online, you'll find a huge amount of kind of sketchy material because it is slightly hallucinogenic, apparently. So there's a lot of material kind of from the drug side of the culture about Scaledium and not a lot on cultivating them, but some of them are quite lovely plants to grow in the greenhouse. Prenia is another genus in the Mesemb. A lot of Prenias are kind of weedy. Prenia sledeniana is kind of an interesting plant. It's never been tremendously long-lived for me. It often seems to go downhill after it flowers, but I have tried cultivating it. So the other subfamily in the Mesembs is the Ruchioideae, and probably the largest genus in the Ruchioideae is Ruchia itself. And it's not particularly well known. I'm definitely not a specialist in Ruchia, but it's maybe about 400 species. It's quite a large group. And a few of them are kind of neat things like Ruchia crassa here, which may be worth considering for growing in the greenhouse. A lot of them are fairly large shrubby plants though, and probably kind of unwieldy in a situation unless you've got a climate where you can grow them outside. Mitrophyllum here is, so Mitrophyllum is a genus of plants that have dimorphic leaves. If you look really closely here, there's a kind of a pair of large tongue-like leaves, and then also a more conical leaf coming out of the center of each of those pairs. It looks a little bit like a bishop's mitre, I guess, and that's how the genus got its name. And it's part of a very distinctive group that has this kind of morphology with the two distinct types of leaves that it produces over the course of its growing season. And they're all very strongly winter growing plants from Namaqualand in South Africa. Another Mitrophyllum is one that probably doesn't have a name yet, but kind of a neat plant, maybe related to Mitrophyllum grande in the last slide. One interesting thing about Mitrophyllums is that in cultivation and probably in the wild too, they take an extremely long time to grow up to maturity. So this is probably almost a 20-year-old plant that's only been flowering for the last couple of years. And that seems to be true for growers all over the place, that it takes probably 10 years at least to get a mature Mitrophyllum of most species from seed. A couple of species are maybe a little bit faster than that, but it takes a long time for them to reach their adult state where they're actually flowering, sort of regardless of how well you grow them or how much sun you give them. Mayoriphytum is another genus in the Mitrophyllum group, and here it is in habitat. Dicracolin is part of a number of genera that used to be thought to be closely related to Mitrophyllum and used to be included in this informal Mitrophyllum group. But I think especially with the modern phylogenetic or evolutionary work involving DNA sequencing, Dicracolin and a number of other genera have been taken out of the Mitrophyllum group and seem to form their own little group that are morphologically somewhat similar to the Mitrophyllum group plants, but apparently unrelated. And then its related genera actually come out as evolving fairly early on in the history of the Ruchioideae subfamily, and are kind of only distantly related to other genera in that subfamily and form their own quite distinct little group. So that's Dichrocolon itself, and they're small shrubs or subshrubs. Dichrocolon humile here is probably the most attractive member of the genus for pot culture. Monolarias, again, these were thought to be part of the Mitrophyllum group, but turn out to be related to Dichrocolon and not really to Mitrophyllum at all. So monolarias here in the northern parts of the Knerslachta is where this particular plant are very strongly winter growing. They're totally dormant and look very dead. Spring up when they get moisture in the fall. They produce these leaves with sparkly bladder cells all over them. And right in the middle of winter, most of them flower, and kind of the coldest part of the winter, they produce often quite large, spectacular flowers. A little bit difficult to grow like this and to get them to flower in cultivation. It's, I think, maybe hard to give them enough sunlight at the relevant time of the year to get them to grow well like this and to flower. Uophytum is a little genus, also in this Dichrocolon group, so related to Monolaria and Dichrocolon. And this is like Dactylopsis, another plant that's endemic to the Knerslachta, doesn't grow anywhere else, and often quite abundant in that part of South Africa. This will form colonies of thousands and thousands of plants, just covering every inch of the soil. Soma is a little genus of 2 species of Dichrocolon relatives, formerly thought to be part of the Mitrophyllum group, but it turns out not. And they're short-lived perennials. They probably, in cultivation and probably also in their natural habitat, they don't live tremendously long. So you've got to restart them from seed periodically. But they've got these amazing bladder cells all over the surface of the plant, and they die down to just a tiny little button-like structure at the soil level during the summertime. They go totally deciduous and spring up again in the next year, at least for a couple of years. This is the other species of Diplosoma. Diplosoma leucophyae, the last one. This is a Knerslachta plant that grows among quartz pebbles. Diplosoma retroversum here is, the common name is the duckfoot Mesemb. And this was endemic to an area just a bit north of Cape Town of Renosterveld. So sort of low-lying areas with fairly rich soil. It grew on areas of quartz pebbles that were sort of devoid of a lot of other vegetation in the area north of Cape Town. A lot of the habitat where these guys used to grow has been converted to winter wheat cultivation. This area for Renosterveld north of Cape Town is kind of the breadbasket of South Africa. And so it's a fairly endangered plant. Probably a lot of its former colonies have been wiped out by agriculture. And a funny case in the Mitrophyllum group now, but used to be thought to be possibly related to Monolaria. This is Monolaria globosa, or Merophytum globosum, an odd plant from the area of, it's just endemic to a small area in sort of coastal Namaqualand. Thought to be a Monolaria originally. It never really fit into that genus very well. More recently transferred to Merophytum, which it may fit in a little bit better. And the DNA studies do put it fairly firmly into the Mitrophyllum group, rather than as an ally of Monolaria in the Dichrocolon group. So that's probably where it's wound up. It may actually need its own genus. It doesn't really fit in with Merophytum very well at all either, or it might actually be considered to be a Mitrophyllum, but sort of a messy situation taxonomically. Gibeums. I think Catherine Arthur is going to talk about gibeums a little bit more later on, so I won't dwell on them too much. This is in the Spheroid Institute in Stephen Hammer out in California. I can't really get them to grow like that. But my favorite gibeum, and probably a lot of people's favorite gibeum, is Gibeum hortensii, which used to be its own monotypic genus Muria, but I think generally considered a gibeum these days, and probably correctly so. But this is the shmoo plant, is the common name for it. Let's get them to grow like this. They need a huge amount of light in the wintertime. It's another genus like that that tends to be a little bit tricky in cultivation. Let's see, Gibeum hortensii. Here again, this is in flower, and maybe not the best developed flower in the world, but I was kind of proud of myself for getting them to finally flower after many years of being sterile and not blooming at all. There is a hybrid of Gibeum album crossed with, which is maybe a little bit easier to grow than straight Muria. Eudentaria is a genus of Dracophyllus from coastal areas in southern Namibia and northern South Africa. So it's a coastal plant. The coastal regions of South Africa, by the way, probably never experience frost, so it would be especially dangerous to expose your Eudentarias to especially cold temperatures. Jensenabatria is another coastal plant, kind of related to Eudentaria and Dracophyllus, but a monotypic genus. This is the common name is Jensen's grape. So it has these delightful round leaves and a little kind of trailing shrubby stems. And here's some sort of ideas for a slide that I stole from Ernst van Jaarsveld, and he's responsible for these slides. I've never been to visit Eudentaria. It's from a very isolated area in Namibia along the Atlantic coast. It's from one little speck of rock sticking out into the South Atlantic called Dolphin's Head, which is sort of isolated with these dune seas in the inland parts. And so yeah, it just grows on that little rocky area out there and maybe a couple of adjacent rocks. And Ernst had an opportunity to visit these plants in habitat and took some photographs of the same plants that had been photographed in the 1970s. So a little bit more than 30 years later, and a lot of the plants had hardly changed at all. So you can compare these two photographs of the same plant in habitat. In cultivation, it seems to appreciate more moisture, sort of more reliable moisture than a lot of winter Mesemms. And I think that's because of its habitat where it grows naturally. It receives a lot of fog, and probably in spite of living in this very barren environment, it does receive fairly regular moisture from fog and condensation at night, and doesn't like to dry out too much if you're growing it in a greenhouse. Argyroderma is a small genus that's entirely native to the Knurslachta region. So it's quite abundant in the Knurslachta, but not found anywhere else at all. But charming little plants tend to flower in the sort of in the autumn or the middle of the winter, depending on the species. Antimima dualis here is another Knurslachta endemic. Antimima is a big genus. A lot of them are kind of scraggly shrubs, which not many people grow. But there's a couple of species that are interesting as potted plant subjects, especially Antimima dualis here, which looks a little bit like an Argyroderma, sort of like a small Argyroderma. But Antimima is kind of related to Ruchia, and a lot of Antimimas were formerly placed in the genus Ruchia. Caridopsis is another genus of strictly winter Mesemms, strictly from the winter rainfall parts of South Africa and Namibia as well. And one of the neatest ones to grow is Caridopsis peculiaris here. Caridopsis has a little bit like Mitrophyllum, it has dimorphic leaves. So it's got a kind of a flattened pair of tongue-like leaves. And in the center, it produces a more fused pair of leaves that'll form a sheath over the next pair of leaves during the summertime and protect the next winter's leaves inside as a dry sheath over the summertime. Caridopsis has very complicated seed capsules. The seed capsules have a lot of membranes and baffles, which kind of prevent the seeds from being dispersed too early. They kind of hold on to the seeds a bit. This is the habitat of Caridopsis peculiaris here, and it's a very strange habitat near the town of Steinkopf in Namaqualand in the Richtersveld. And here's somebody looking for them out in their habitat. The ground that they grow on is covered by broken, kind of dark colored shale, and almost nothing else grows in the spot where Caridopsis peculiaris grows. It's, aside from the Caridopsis, it's almost entirely barren. And here's one of the plants in its habitat. And it must be a tremendously dry, hot environment, especially in the summertime. I imagine those rocks really heat up in the sun. Caridopsis pilanzia. This is a big plant. This flower is probably about the size of a teacup, and blooming right in the middle of winter, sort of right in the dead of winter is when it flowers, if you can get enough sun to get it to flower. Fenestraria is a coastal plant, and it grows in old stabilized sand dunes. So it grows in northern, kind of the northwest corner of South Africa and in southern Namibia as well. And it grows in sand, and it's a really good example of a window plant. So these leaves have sort of a lens-like top to them that's translucent and lets in the sunlight. And it would be buried right up to the tops of the leaves if it was growing in its native habitat. It's kind of hard to get them to grow like that, even in a sunny greenhouse in cultivation. Yeah, these leaves will be exposed. The window at the top will be exposed during the winter growing period. During the summer, the leaves shrink a bit, and it kind of hides back down into the ground and disappears entirely during the warmer months in its habitat. Didymeotis grows in extremely arid areas in the Tangpo Karoo, and they're slow, and it's hard to get them to flower in cultivation, and sort of a tough thing to get looking decent in cultivation conditions. But yeah, it needs a tremendous amount of light and very careful watering if you want to try and keep your Didymeotis alive. Van Herdia is a small genus. It comes kind of from the eastern parts of Namaqualand, kind of the boundary area between Namaqualand and the Bushmanland area to the east. And so sort of on the boundary of Namaqualand winter rainfall regimes and Bushmanland, which gets more kind of spring and autumn rain and thunderstorms maybe in the summertime if they're lucky, but comes from an arid area and kind of irregular rainfall, but tends to grow more in the winter in cultivation. Van Herdia primosii looks a little bit like a lithops, and it does have windows in the tips of the leaves, but it's a lithops with little teeth on the edges of the leaves. Kind of a neat thing. Glotophyllums are from the Little Karoo, and they're fairly easy plants to grow, but kind of tough to get to look as colorful and as compact as this, at least for me with my cultivation conditions. This is a photograph from California from Stephen Hammer's collection. I have a really hard time getting glotophyllums to look like this. Glotophyllum pearsii here, again in Steve Hammer's collection, not in my own. Glotophyllums are sort of getting out east, kind of at the boundary of the winter rainfall regime area. And they're a little bit opportunistic in their growth rather than being strictly winter growing sorts of plants. They're evergreen, they keep their leaves year round, and they will respond to water pretty much at any time of the year. So look at these plants in the spring when a lot of them are kind of going downhill, but some of them are really at their peak. Drosanthemum here with bladder cells in the surface. And Drosanthemum is a genus of kind of shrubby winter mesembs, and they tend to put on their flower shows. They flower in mass in the springtime. So they put on these spectacular shows in the springtime, and they're grown as bedding plants it's like popular as bedding plants. They're the sort of ice plants that the people in California plant in their highway medians and that flower nicely in the spring. And this is another shot from Southern California, showing these plants being cultivated as just a garden as a ground cover. Dorothianthus is one of a number of genera of winter-growing annuals in the subfamily Ruchioideae and the Mesembs. Dorothianthus are sometimes sold in garden centers as an annual. In my sort of climate, I have a really hard time growing Dorothianthus. I think it's probably hard for me to give them enough sunlight to get them growing in the winter time and get them ready to flower in the springtime, which is how they would in their native environment. And this one in particular is a spectacular flowering plant and a spectacular plant in flower, but it's kind of difficult to cultivate. I've never successfully cultivated it myself, but Dorothianthus workii has got these great flowers, but even the seeds are quite difficult to germinate, and it's a hard plant for me to grow. Ethophyllum is another small genus of a winter-growing annual. Ethophyllum and Dorothianthus are sometimes included in the genus Chlorydum. So sometimes all put together into a single genus of winter-growing annuals. What am I doing on time? Keep on going for a little bit. Lampranthus, shrubs with large flowers, winter growing, flower in the spring. And some of the compact mesembs put on kind of a color show in the spring. Some of them develop interesting colors in their leaves when it starts to warm up and they're getting ready for dormancy. Here's Conophytum monii, showing its spring colors right before it goes to sleep and goes dormant for the summertime. And so summer, here's a very sad looking Diplosoma going dormant for the warmer months. And Mitrophyllum with those miter-like leaves dried up to a protective sheath to cover the leaves until autumn. Codophytums looking very dormant as they do for about 6 months of the year. But even the conophytums, these sort of classical winter-drying mesembs, there are a few species that put on a nice show in the springtime or in the summertime even. So here's a Conophytum minusculum subspecies Istiflorens, flowering right around the summer solstice. And similarly for Conophytum frutescens here, it's growing. You think of conophytums as being totally dormant in the summertime, but some of them, especially species like this, grow a bit and flower primarily during the hottest time of the year. So even in the summertime for our winter mesem collections, there's often a little bit going on. And so I think I'll end it right there, maybe going over a little bit. And I don't know if we have time for questions. I will turn off my screen sharing for now.
Thank you very much.
Thank you very much, Matt.
Oh, you're welcome.
I don't really think we have time for questions now.
Okay.
So can I just really thank you very much for your presentation. I was interested to see how your cultivation techniques compared to ours. And actually, apart from some slightly different materials, they're obviously very similar. But what very few people in the UK have any experience of is growing under lights. And quite often, I do act as a sort of conophytum agony uncle, and I'll quite often get questions, particularly from the Far East, about growing them under lights. So it's very interesting to see that you do it and that it can be done very successfully. Thank you too for the sort of run through of the 4 seasons. And I was thinking to myself, well, yeah, your plants look very similar in Connecticut until you started getting to a few things, such as Dactylopsis, which we find almost totally impossible. And things like diplosoma too, they're very, very difficult under UK conditions. So we very much sort of admire you for, growing those there. So anyway, can I thank you very much on behalf of everyone who's watching. And Jennifer and I would just applaud you on behalf of the other 200. So thank you very much.
You're welcome, and thanks for having me.
Yeah, thanks. I think Matt was quite happy to stay on a little bit at the end and to answer any questions. So if anybody's got any questions, then we can take those at the end. If you just write them down and hold on to them, we'll get round to them eventually. We are running a little bit later than scheduled now. So what we've done is very quickly reschedule the afternoon. We'll be having a 10-minute break now. We will reconvene at 2:30. We will take 3 of the 5 short presentations. Then we will do another short break and have Robin's talk. And then the final 2 presentations will be short presentations, will be after Robin's talk. So we take questions for Matt and Robin after Robin's talk, and then the final 2 presentations. It does stretch it out a little bit longer than we'd anticipated, but I'm sure anybody that's seen Matt's talk there will realise it's been well worth it not to rush him through and to allow him to finish in his own time. So we'll see everybody back in 10 minutes, and hopefully we'll go from there. Thank you.
Thank you.
Half an hour later than it would have been. Right.
Ian, can I just ask you, are you happy to go at 1:30 as the first presentation again?
If you want, yeah.
Yeah, when we reconvene, we will ask you to go first, please. I can't remember whether it was Alice or Catherine that we said next. Terry and Brian are going to go after the second long talk.
Yeah, okay. I'm happy to go at 1:30, yeah.
Yeah, that's great. Thank you.
Okay.
I shall mute you back again.
I've just posted a link to the lights that Matt uses to grow under. And I think I've sent it to everyone. They run about $50 US and they work great. So if anyone were to ask like what lights we use, that's the light we use.
I have allowed people to unmute themselves if they're interested. This is John. You could talk amongst yourselves during the break. There's no reason not to do that. So you should be able to just click unmute and talk.
Can you hear me?
Yes.
So I just posted a link in the chat, which is the Amazon link for the lights that Matt grows under.
We heard it the first time. We heard it the first time.
Oh, okay. All right, good.
You were unmuted because you were already your computer there is already a speaker computer. So.
All right. I figure since I heard you say that a lot of people don't go under lights, a lot of times the first question will be like, well, what lights should I get? And this is a $50 light that works great. And so that'll save a little time and if they ask Matt about going under lights.
There's a lot of options if you start looking into it, though. It's a little bit overwhelming, maybe, but these seem to work okay. I'm sure there's plenty of others that would work as well.
We've grown multiple seasons under them, and the plants flower, and the plants look really, really good.
Yeah, sorry, I'll just come back in. I know you were talking about growing under lights. I keep all my dwarf aloes and aloe hybrids. I actually grow them under the bench on a heated sand bed under Growlux tubes, and they flower and they look brilliant. And it also extends the day length in the winter. So there's another angle.
Yeah. Maybe people could all post, you know, what lights they use in the chat, and we could have a comparison, because there's just so many options nowadays. And it takes a really long, you know, at least a growing season to determine if you like the light or not. So it's great to hear from other people, you know, I have tried this and it works well.
I've just started growing the lights this year for the first time. Well, I changed that. I was actually used a fluorescent T5 tube before to actually grow seedlings, but this year I'm actually working with LEDs. So I've got two types to try at the minute, and I've actually got them in the greenhouse now. One of them was sold by IKEA, which is like a bulb, an LED bulb. And the other one was like a T5 strip light, which has come from China. So I've got both trialing at the moment. The IKEA bulb is actually positioned above my conophytums just to supplement daylight. And I'm finding, so it's actually out in the greenhouse, and I'm finding that I've already flowered 2 conophytums I've never flowered before at sort of 9.5 years old from seed and 11 years old from seed. So I think potentially there's a lot of scope for it. But it's still trial period for me at the minute. So looking forward to better results coming up through the season.
Yeah, you can go away on vacation and not worry that the power is going to go out and everything's going to freeze, or your, you know, vents aren't going to open, everything's going to cook. Like, growing under lights has disadvantages, but also has like that safety advantage.
I think the worry for me with them being in the greenhouse is that because obviously once the light drops, once the light level goes down, it really is a beacon, and I worry that I'm going to get visited by the police or maybe sort of inquisitive neighbours. So I tried to put them on a timer for the natural daylight as well, but even so, it's improving what's available to me because of cloudy weather. So it seems to be positive. And now the technology with LED is so little draw of electricity. It's current. It's amazing. Yeah.
We wrap mylar around the shelf that we grow on, so it kind of reflects the light back and it doesn't make the room feel so weird, you know, that horrible glow. You might, I guess in a greenhouse you can't really do it. And inside you can put mylar facing inside with the reflective part inside the window. That might reduce the glow in the window, but probably not with a greenhouse.
For the seedlings, I actually created a wooden box which was light tight, which was lined with reflective material, which was perfect. I could actually have sort of 18-hour daylight through the winter and everything, but to actually illuminate the bench, that's it. And I've got bubble plastic up as well, you see. So the bubble plastic actually receives the light and creates this massive balloon effect, you know. So it's quite dramatic. So yeah, so I avoid too many prying eyes, I think.
Wow, 18 hours. That's a long.
That's an excessive. That's for seedlings. It was really cacti and things, you know, to emulate sort of like 1 month, you know. But wouldn't do that with my Mesemb, you know.
I've heard of people doing that for winter growers, even though just for seedlings to you know, keep them under very long day length, and apparently it works out for at least a while. Before it just causes them to go dormant, no matter the time of year, if you're giving them a very long photo period. You know, definitely not something you can do with adult plants, but the seedlings I think are more adaptable and maybe might benefit from that even. I haven't messed around with that much. I just sort of keep my lights at 12 hours.
Well, there's obviously a lot of trial and experiments to be made in length of time that the lights are on as well. So yeah, I think it's exciting times ahead with the way technology is changing now for that. Yeah. And two kind of items within a month of switching the lights on that I've never flowered before is a good start, I think.
It's so easy to try. You can get a small light for reasonable money and just, try it with a few pots. You don't have to give up your greenhouse or give up your other methods. You can just see if you like it.
Well, the actual IKEA bulb light is just in a Edison screw fit. And so you just wire that up above 25 centimeters above the plants they recommend.
Right.
So you get a spotlight. So I've got a small tray of 10-year-old Moanii seedling plants, not seedlings anymore, that have never flowered, and I'm hoping that they're going to do it within the next month or so. But fingers crossed,
Yeah, it's just about that.
Brian, is it expensive to run the lights?
It's only 10 watts, so really it's expensive in any hobby, isn't it? But as far as when you think about it's less than the fluorescent tubes. So
right,
the
thank you.
Yeah, so it's not a lot of expense really.
Okay,
and
thank you very
to a
much.
greenhouse, it's really cheap. We have plants we've grown from seedlings under light to flowering size, and they've barely received any natural light, like a little incidental light from the window, but you really can go through the whole life cycle under lights. And sometimes it helps to a little bit underwater the plants under the lights if they're not getting as much sun. And you water them the same amount as the greenhouse, they'll etiolate. So you have to learn a cultivation technique under lights, which is a little bit different in the watering. And you want a really, really lean soil. Don't put any organic matter in there because, a little extra nutrients, a little extra lights, water and nutrients for plants under lights can have a really extreme effect that it wouldn't have in the greenhouse.
Right, thank you. It's now past again, so I think we're almost ready to go again. We've got a slightly changed program, as we said before, so anybody who is just joining us, because I noticed the numbers are going up and down slightly from time to time. We're going to take 3 of the short presentations now, and then the second major talk, and then 2 of the shorter presentations after that. I think the first one to go is Ian. So Ian, I don't know if you've got the ability to share screen or not at the moment. You should do. He has. So I shall hand over to Ian.
Sorry, I muted you for a second, and the speakers are going to need to unmute themselves. I had to mute all.
Sorry, I don't know where I got to there, but Ian's one of our regular showers at the Northwest Mesemb show. He lives in North Wales, has grown Mesembs for a long time, has visited South Africa. So whenever you're ready, Ian, we shall let you carry on with your presentation. Have you unmuted? We can't hear you. I've lost you on the screens.
Dear.
Are you there? Can hear you now, Ian.
Can you hear me? Right.
Yes, I can now.
And can you see my screen?
I could see your screen, yes.
Brilliant. Okay. Here we go. Yeah. The short talk, Some Plants from the Southwest Africa, presented by Ian Robinson. It's me. Okay. All these plants are grown in my greenhouse. Argyroderma is grown in northwestern Cape and southern Namaqualand, where they get winter rainfall of less than 100 meters. This is where we are. Sorry, 100 millimeters, yeah. There's Argyroderma testiculara, which comes in a variety of colors. As you can see, we've got in my greenhouse, a purple one and a white one. And there's also a plant from south of Bitterfontein that I have, which I think is probably also testicularia, which is a yellow flowered one. These photos are all taken early 2006. And then this is the same plant as the one in the top corner, taken in September 2020. So you can see that's 14 years it's managed to produce the second head. I'll talk more about this scarring later on in the presentation. They can be very slow. This photo was taken in 2002 from seed in 1977. So it's 25 years old at that point. It came under the name originally to me, the seed as ovale, is known more now as Pearsoni or even testiculari variety Pearsoni. You can see it's got 6 heads. In 2020, we've got the same plant, which looks almost identical, except that the stems are longer. Difference in color is actual. In 2002, it was looking quite blue. This year, it's looking much more silvery, as a habitat plant would look. They don't always grow as you might like them to. This one is, again, it's a testiculari, which had a deformed head, which went blind. And I wondered what would happen. And in fact, that head just didn't regenerate the following year. Split bodies. Well, this is probably down to bad watering. I work on a policy of looking at the current growth that's happening. And if the current growth is turgid, not wrinkly, then it doesn't need water. If the old growth is wrinkly, ignore it, doesn't matter. But if the new growth is wrinkly, then you can water. Now, these two do seem much more prone to splitting than some of the others. And they're growing right next to and getting the same watering regime. They were in 2-inch pots, same watering regime as these other plants, which are not splitting. This is Crateriforme and Alba, about 30 years old in a 9 centimeters pot. And Mesemb, again, a 30-year-old plant in a 5-inch pot. These smaller headed ones seem to grow a lot faster than the bigger headed ones. And a congregatum, and this again taken when it was 23 years old. It's got a few of these marks on, which may be the marks that Matt was referring to. But here we are at 30 years old, no marks on it, same plant, and a few more heads. Okay, moving on to southern Namibia and the northwestern Cape with light winter rainfall, Lapidaria margaritae. I've found it comes in a range of colors from a pale bluey color, but more often to a pink or browny color. And this same plant, photograph taken in 2002, with no nasty marking on it, in 2020, looking much paler in color and more towards the brown end of the scale. And you can see it's got one bad leaf here. That's not bad. I find they mark incredibly easily. This one you can see has gone through a very bad patch, and this one even worse. And there's not a lot that I've found I can do about this. I don't know if anybody else has the answers, but because they hold on to their old leaves, then it's very difficult to get rid of the marks once they're there. You just have to wait for the growth to grow out and get new heads appearing like this one, which is quite good. And I'm hopeful that eventually it might be okay. So Gibeum, growing mainly southwestern Cape down here at the bottom, mostly experiencing both summer and winter rainfall. And we have plants such as this, Gibeum dispar. In 2002, it was in a 7-inch pan. I got the plant about 1986, and it was already quite a big plant. And it's now, I think, in an 8.5-inch pan in 2020. It does, given getting hot conditions, get these brown, and I'll go a bit closer, brownish looking marks on it, which worried me the first time I saw it. I think it probably is heat stress, but it doesn't seem to matter too much, and it does grow out of it. Another plant which I've lost now, again, a very old plant, it's pubescens, beautiful looking plant with almost furry leaves. And you can see this is an old plant. I flowered it once when it was probably about 35 to 40 years old. So there must be something I'm doing wrong, except I managed to keep it growing for a great length of time. Great pity, it resented being repotted into about a 10-inch pot. And that was the end of it. It just gradually went backwards. One of my favorites, Gibeum album, and again, remember these get winter and a little bit of summer rainfall. Most definitely Gibeum album, if you don't treat it as a winter grower, it just gets smaller. Since treating it as a winter grower, I've managed to get it to grow better and finally flowered it. The name album refers to the plant body color, and it does have flowers varying from white to pink. So it does vary. Dinteranthus also grow in the Western Cape and up into Namibia and Namaqualand. And I'm just showing you one plant, and this was the first plant I had, which is Dinteranthus puberulis, which I find is the easiest of all the Dinteranthus to grow. It grows pretty quickly. This one is white to pink flowers. So when I found this one with the yellow flowers, I was really excited and thought I had something rare. And then, of course, when I looked at the literature, I found it more normally has yellow flowers, and it's the white to pink version that had appeared much later on. And my final slide, Kiriodopsis, grows down from Namibia all the way down Namaqualand to South Africa, the Western Cape. So it will get winter rainfall. And this is just a couple of slides. Matt already spoke about this, but these are in 2-inch pots. They're about 18 months old, and one of them is growing pink and the other one is growing green. And that is the subject for another talk. I've got a great theory about the pink and the green, and that's the end of my 10 minutes.
Thank you, Ian. Brilliant. We shall save the pink and the green for another day, shall we?
Yeah.
Okay. I think really time is pressing. We just need to press on. I just thank you at this stage for that. And again, if anybody has any questions and Ian's still about at the end, we can always come back to that. I think it's really to ask Alice to unmute because you're still muted, and then you can go whenever you're ready, please. Alice has been a long-time member of the MSG and has extensively toured Southern Africa and an excellent cultivator of plants.
Yeah, I've just lost it again. Okay, can you see it?
Sorry, I've just come out. Yes, we've got your first slide, Alice. Yes.
Right, okay. Well, mine's just a random selection of plants from my greenhouse. And I'm starting with this Grotiphyllum, which I must admit I feel a bit ashamed of now I haven't seen Steve Hammer's. But this is probably one of the nicest ones to grow. It has these nice grey leaves and rounded bodies and nice yellow flowers. Well, most of them have yellow flowers. This particular plant originated from Rick Marshall, who many of you will know, who was a superb Mesemb grower and who specialised in conophytums. This plant is now growing in an 8-inch pan. My second plant, Titanopsis calcarea, I grew this from seed in 1992 from MSG seed, and it's now in the 6 1/4 inch pan. Flowers well every year, and There's 3 species. This is Calcarea, Schwantezi, which includes Primosi, and Hugo schlechteri, which is probably perhaps the most attractive, with these sort of chocolate brown tips and this sort of greyish blue body. Semophorus, I'm quite keen on these plants. I do have a bit of problems with brown markings on them. This I have labelled as Herrii, but evidently that is now longifolia, although my other plants of longifolia have much larger leaves. Semophorus have a sticky epidermis, so in habitat the sand sticks to the epidermis and gives the plant very good camouflage, makes them look quite brown. This one flowering, I think, in about November. Tanquana hilmeri. Tanquana used to be classified with Pliospilos and was split off by Heidi Hartmann in about 1986, I think. There's 3 species of Tanquana, Hilmeri, Archeri, and Prismatica. They're very prone to splitting, so you have to be extremely careful with the watering can. It's okay to give them a reasonable douse when they first come into growth, but once they're flowering, you have to be extremely careful. Nellia. This is a northern species. It's named after Professor of Botany at Stellenbosch, G.C. Nell, who also did the monographs on lithops and Gibeum. There's 2 species, Pilanzi and Schlechteri, and I think this is Pilanzi. It was grown from MSG seed in 1994. And it sometimes produces more than 1 flower. You can see the pedicels here with just 1 bud on, but some over here have 2. Evidently Schlechteri is always single flowered. Pilanzii does tend to produce more than 1. The interesting thing about this group of plants is that the flowers, once they're open, they remain open, unlike most Mesemb flowers, which only open in the afternoons. Here's my lapidaria, also has brown marks, which, as Ian says, extremely irritating. I don't know why it occurs, but you can see them in here and on the edges of the leaves. This plant's growing in I think it's about a 6-inch pan. No, it might be a 5-inch pan. It's a little bit embarrassing because this plant actually originates from a plant I bought from Ernest Hepworth in 1970, which makes it 50 years old. So yes, it's a very slow grower, at least in my cultivation, but it does produce lovely yellow flowers and well worth growing. Deuteranteria, Covis montana, this is also known as Simpsoni. It's probably one of the easiest Deuteranterias that you can grow. It flowers almost sort of continuously through the summer, producing 2 or 3 flowers at a time, and it has these lovely dentate margins. But this does have a lot of marks on the leaves, which does rather detract from it. Chirodopsis. This is a plant that I received from Gordon Rowley just about the time he was giving up his collection in 2016. it was a huge plant which I had to sort of take cuttings and trim into some sort of shape, but it's done quite well. It produces flowers each year, but it's not overly floriferous. It, as you can see, we've got 3 flowers here. And this is how it tends to flower, with rather crinkly petals. Maybe this is just because it's not fully opening. It could just be a light problem. But I do have several plants of this from different sources, which all tend to do this. Schwantesias, I grow a lot of these. They do well up on the top shelf of the greenhouse in plenty of light. This particular plant is an interesting one. It has these sort of spoon-shaped leaves and these lovely large yellow flowers. There's 11 species according to the handbook, and I must admit, although I've got several plants, I don't know that they've all got species names. And finally, I have to finish with the conophytum, and this is a conophytum that I received from Derek Bowdley shortly before his death. It's growing in a 9-inch pan, and is certainly a very large plant, but unfortunately I have no idea of how old it is or its history. So yeah, so that's a bit of a mystery. But Conophytum perianthus, subspecies Herianthus, is a plant that hasn't been found in habitat for some years now, probably over 20 years, first discovered in 1925 and obviously named for Hans Hare. But it's very distinctive with its sort of marble-like bodies and these sort of sharp edges to the bodies, as opposed to its sister species, if you like, subspecies Rex, which is a green-bodied plant with red edges to the bodies. Here it is in flower, so you can see the flowers. It flowers quite well. In fact, I think usually it flowers from every head. So that's my 10 Mesembs for you.
Thank you very much, Alice. That was great to see. Thank you. I don't think we really thought it was going to be such a rush and we wouldn't be able to thank people properly after each one of these presentations, but I think we need to move on, if that's okay. Thank you for your bit. We need to find Catherine. Catherine, can you either put your hand up? You there? I've lost you. I can hear you but not see you. Are you ready to go to screen share?
Yeah, okay. I shall go from here.
Yeah, okay. Catherine, oh, there we go. Yeah, Catherine's another one of our long-term MSG members who's currently living in southern Spain. So has got a slightly different approach to cultivation than the much colder climate that we tend to get in, certainly in Manchester and in the rest of the UK. So, Catherine, thank you when you're ready.
Okay, thanks, Ivor. I'm going to cover just 3 quick topics. I'll just share my screen with you and switch off my video, actually. That for a minute, sorry. Okay, can you see that?
Yes. We can hear you and we can see the screen.
Fantastic. Okay, I love gibberiums and I really love growing gibberiums, but I know I'm not the only one who struggles to give them just the right amount of water, to keep them looking good. I grow most of the 18 or so Gibeum tacta from seed, and a few of them are shown here. Making sure that they have enough water to flower, but not so much that their leaves stack up or crack open, is not the easiest of tasks. And since it's such a widely diverse genus, no single cultivation method's ever going to suit all the species. So among most adult mesems, the smaller the leaves and the fewer the branches, the less water the plant can tolerate. So it's no surprise that the smaller compact Gibeums are much more sensitive and need far less water than the larger leaved or shrubbier forms. And as well as body size and growth habit, skin type also gives a good indication of how much water a particular mesem can cope with before it completely self-destructs. And Gibeum species essentially have one of three basic skin types. So a velvety skin, a more densely pubescent or hairy skin, and a smooth glabrous skin. And each of these epidermal types has a very different sensitivity to moisture. Each of them responds in their own separate way to drought or to excess water. Now, the thin-skinned velvety species, represented here on the left with dispar, they're the most tender, and their leaves soon split if they get too much water too fast. And not enough water, and the leaves feel soft and squishy, and they'll eventually begin to shrivel and dry up. The thicker-skinned pubescent types, like album here in the middle, and the smoother-skinned species on the right, they generally have much greater resistance to overwatering, and they can more easily stretch to accommodate water. And when they're thirsty, the glabrous types tend to wrinkle, whereas the hairier bodies, as Ian found with his album and pubescens, they'll simply tend to shrink. Most growth forms within Gibeum are represented by each of these three basic skin types. So that includes these shark's head forms with their extremely unequal leaf lobes, or these bigger bodied boat-like, or what I like to think of as beak-shaped forms that have a large curved upper leaf lobe overhanging the smaller lobe. So what I'm saying is that even when the species are similar in size and they have similar growth habits, they can still have massively different watering requirements. And it's their skin type that can help us work out which ones are most split prone and which ones definitely need heavier drinks. A quick note about flowering, and unlike I don't include Muria here, although if I did, it flowers at the same time as album. This calendar kind of shows the timing of my plants in flower here in the northern hemisphere, and only November's John Stoney is not one of my plants. I don't yet have a flowering specimen of that, but like Conophytum, Gibeum have a very long flowering season, which begins with Nebrownie late in September or early October here, and ends in June with Gibeum album. And knowing when a species flowers can help us work out an appropriate watering regime. So it can guide us in the frequency of watering over the year, as opposed to the quantity of water the plant might require each time it's watered. So my Gibeums all get a fairly good drink in late August or early September, about the same time that I gently wake up my Conophytums and my other winter growers. And after that, the Gibeums, any that haven't flowered yet, just get a few little top-ups over winter to keep them in shape. But one by one, I begin to withhold water once a plant has flowered. And for the rest of the season, they more or less survive on heavy dews and any moisture that's there, which isn't much here, whilst they get on with replacing their old leaves with the new ones.
Catherine, sorry to butt in.
Yeah.
But have you changed your slide? We're all still seeing the first slide you showed. Is that correct?
Oh, you're joking. No.
Oh, we're just seeing the Gibeum dispar slide, the one with the 6 pictures on.
Oh, you're kidding. Okay. No, there's plenty more.
If you come out of screen share and go back in, that might solve.
Oh, I'm sorry, guys. How's that? What are you seeing now?
At the moment, nothing yet. Now we're on the 3, Knee Brownie, Pilosum and Heathii.
Okay, I'll quickly cover that again, just to say that these are the compact mesems that are much more sensitive and need less water. Can you? Oh, I'm going to have to. Sorry, guys. What are you seeing now?
A brownie eye, Pilosum and Heathii.
And now, same?
Yes.
It's not working on my, I'm going to have to, dear me.
Skin types we can see now.
Catherine, I've had this problem in PowerPoint. If you go back to the basic slide sorter, you may be able to work it from there.
Okay. How's that?
Seems to be updating.
Okay. Okay, I'm going to just run through. These are the smaller, most compact ones. These are the skin types, so the velvety, the pubescent, and the glabrous types that vary in their moisture holding capabilities. Here we've. Can you? Is it moving for you now?
Yes.
Yes.
Okay. Okay. Here we've got the 3 basic skin types, the small ones, the velvety, which splits very easily, Gibbeum album, which holds water much better, as does Petrense, and they'll they cope with far much water before splitting than the Gibbeum dispar. These 3 are the shark's head types, and they have the extremely unequal lobes, but essentially have the same skin type, so the velvety, the pubescent, and the glabrous. And these are the bigger bodied boat-like, or the beak-shaped forms with the overhanging lobes. And basically their skin type can help us work out which ones are most split prone. This is the calendar that I was talking about, my calendar of flowering here, and that knowing which ones flower when helps us to work out the frequency of watering. And mine get watered in late August, September, and I begin to withhold the water once the plant has flowered. And none of them flower in the hottest, driest, brightest time of the year. So July, August, September, nothing's in flower here. They're essentially kept dry and they're dormant here. And a final note, the 3 ecotypes that have white flowers as well as pink ones, so that's Heathii, Velutinum and Album, and they're all shown here. They all flower towards the end of the season, so in the warmer, brighter months. Okay, apologies for the disaster there. I'm going to move on to Secandra serrata. So this is the only species in this genus, so it's a monotypic genus, and the plant's quite rare in cultivation still. It's not a new discovery though. It was actually described more than 250 years ago, but it hadn't been seen either in habitat or in cultivation, so it was presumed extinct. And in 2005, it was rediscovered at its only known habitat northeast of Cape Town. Stephen Hammer was given some seeds, and he saw his single plant flower about 4 years ago. And some of you may recall reading his notes about it in the bulletin. Fortunately, the plants are self-fertile, and the fruits from his plant contained enough seed for a limited distribution. And 3 years ago, Terry sent me a packet of these seeds along with my MSG seed order, with the note that they'd be tricky to germinate, and he hoped my hot conditions here in Spain might overcome the known germination problems. I sowed the seeds pretty much straight away, and by the happened, which I wasn't at all surprised about. I let the pot dry out. I put it on a shelf in the shade house, and I essentially left it to bake in the summer sunshine to get wet whenever it rained. And a whole year and a half passed. Nothing at all stirred in the pot until suddenly, a week after we'd had a big thunderstorm here, 7 little seedlings magically and simultaneously appeared in the pot. I did lose one seedling that first winter before I'd had time to work out quite how thirsty the species is, but the rest of them grew fairly steadily and gradually developed the long, waxy, grey-green triangular leaves with the finely serrated keels that are typical of the species, and which in many respects resembles Erepsia lacera, or even an upright Carpobrotus. I transplanted my seedlings into bigger pots at about 3 months old, but they made no attempt to flower that first year. And in February this year, I noticed the first bud forming on the biggest plant. And over the next few weeks, buds had appeared on all 6 plants, with several of them developing multiple flowers. The big golden flowers seemed to take forever to develop, but they finally opened in May. And they're really quite unusual for a Mesemb. The name Circandra refers to the distinctive circle of anthers that you can hopefully see here, which are notably short, and they're arranged in a ring around the top of the floral cup. The flowers only opened at midday, so just for a brief hour or two, but that's enough to gradually ripen the pollen on the anthers. And each day, the pile of pollen that they shed grew bigger and bigger until it completely covered the smooth, shiny, bright green base of the flower. And any receptive female parts, or the stigmas, they were nowhere to be seen, even when I brushed away the heap of pollen. Now my 6 plants, they've been sitting in sun all summer, and the fruits have only just begun to look vaguely ripe. Last week it rained, and a couple of the capsules opened, and there's definitely some nice healthy looking seed inside, so I should soon have some seed for a wider distribution, hopefully via the MSG seed list. Okay, I'll very quickly go through my lovely Chirodopsis denticulata, which I'm lucky enough to be able to grow outside in my garden as well as in my shade house, and it thrives outside in a well-drained rockery, looking its best over winter, especially through February and March when it's in full fabulous flower. The 2 clones that I'm showing here have been grown from seed sown in 2011, and what particularly interests me about this species is the toothiness of the leaves. Now all my clones produce a similar range of leaf types, from the smooth and pointy type to a really quite elaborately denticulate, with a kind of intermediate notched rather than fully toothy form, somewhere in between. If the old pair of leaves only produces a single new pair of leaves in winter, then they'll be smooth and pointy. But when a plant branches and a leaf pair produces the maximum of 2 leaf pairs, then it's the second pair of leaves that's highly toothy. The first pair produced is hardly denticulate at all. So basically, if they get plenty of water over the winter, they're more likely to have the resources to produce those second toothy leaves, whereas in drier years, they may only ever produce 1 smooth or notched replacement leaf pair, and that kind of depends on whether they flower or not. But we really need we need a bit more long-term observation of this species. But my pot-grown plants, my mature pot-grown plants anyway, are certainly slower to branch, and quite often they only produce 1 new pair of leaves each winter to replace the old ones. So they're almost never as toothy as the garden plants. I suspect this may be true of many of the cultivated denticulata we see, especially those confined to pots. And it could well be worth anyone who's growing this species to try giving it a nice big pot and maybe a good autumn watering, if that's possible, to see if it grows any more teeth. Okay, that's it. My time's up. Thank you very much.
Thank you, Catherine. Thank you to all our speakers. It's difficult just to give this the thanks that it needs in such a short space of time, but time is pressing on. It's almost 10 past now. I'm going to suggest that we restart at 15:15, which gives you 7 minutes to boil the kettle and whiz a tea bag round in it when we start back again. And then we can thank all our speakers properly after the end of the next talk. That's if you've not already got a cup of tea in front of you. So I will see everybody back here. I'm going to ask John if he will leave the microphone open and unmute people, so at least people can talk in the next, what's now 6 minutes, I'm sorry. And we will get back and start again for the next part in 6 minutes. Thank you.
So yes, people should be able to unmute themselves now if you'd like to talk.
Yeah.
Well, everybody's very quiet. It's Jim Backus here. I don't know whether people can hear me or not. I wondered if anybody had got any advice on Lapidaria. I was one of the nurseries near Bath, and they were selling a pot of Lapidaria seedlings as lithops, and I put them right on that and bought the plant anyway. I've got about 6 surviving plants, and they do all right, but I feel they could do better. Anybody got any advice? I'll mute myself now.
I would suggest that you water it more heavily and in the winter than you have done in the past. I think the markings that I get on mine are possibly down to not being watered heavily enough. If that's a help, I don't know.
Just like to add my bit about Lapidaria. I struggle with Lapidaria here in Glasgow. But I spoke to Stephen Hammer once, and he actually said you'd water them much more deeply than lithops. Obviously, they are related to lithops to a degree, so he recommended watering them much more deeply. So I still have to do that with mine, but I've got one plant which is hanging on at the minute. That's all I can contribute. I'm sorry.
I went walkabout just now, so I don't know what anyone said about Lapidaria, but I just tend to treat it like a lithops and water it from about May till now.
Can you hear me?
Well, that's what I've tended to do, is follow lithops watering season. So it's interesting that there's advice to carry on a bit further into the winter. So I'll give that a try and see how I get on with that.
Sorry, Jim, did you say your plant flowers?
Yes, I have had flowers on mine.
Do you know when?
They seem to spread over quite a while. They can flower August, September time.
Okay, yeah. Mine are currently in bud, and yes, I'd agree. Put maybe a bit more water up until now, until they flower.
Yeah. Okay. Thank you.
Everyone's very quiet.
It's gone on a long time, I think, already this afternoon. So I think everybody just needs a little bit of a break before we start to concentrate again, don't we? Yeah. I don't think we want to miss any detail from any of the speakers today.
I think with regard to watering, it's important to know where the plants come from and whether they're winter or summer rainfall plants. I have an adage that the Western Cape has a W in it, and that's winter rainfall. And from going east from there, you gradually get more and more summer rain. And that is a guide. How much you give depends on how badly you split them.
Can I just say something about Lapidaria? I don't have any trouble with it at all. I have flowered it, and all I do is water it like a cactus. Oh, it says I'm muted. No, I don't think I am.
Oh, I can hear you, Jonathan.
You can, yes. I just water it like a cactus, which is not quite the same as lithops, of course. So I'm sort of tailing it off now, but I don't fuss and they're fine. If that helps.
Thanks, Jonathan. I'm going to call the meeting back to order because it's now exactly past. If I can ask John to mute everybody, and both Peter and Robin will need to unmute themselves when he has. And then we can continue from there. So, Peter, when you're ready to unmute yourself, I'll let you do
I'm unmuted. I hope Robin's ready.
Can you hear me?
Right, a few words about Robin. She has been based in a cytogenetic laboratory and studied genome size of plants and fungi across a range of groups. She assists and trains staff and students in flow psychometry and is involved in producing data that contributes to a number of projects. Importantly for us today, she is also interested in understanding genome size evolution in the diverse succulent plant family Azoaceae. She has a PhD from the University of the Western Cape, an MSc from the University of Cape Town, and a BA Honours from the University of Pretoria. She has collaborated in a number of projects with Cornelia Clack, J.S. Boatwright and A.R. McGee, and they include nomenclatural novelties in Chirodopsis, a taxonomic revision of Schlepteranthus, subgenus Microphallus, the expansion of Chirodopsis to include Islandfeltia and Odontophorus, and the phylogenetic placement and generic circumscriptions amongst Arenifera, Octopoma and Schlepteranthus. So she has done a lot of study. She's currently working at Kew Gardens, working in the Jodrell laboratory. I don't profess to understand flow psychometry, but apparently it's a technique used to detect and measure physical and chemical characteristics of populations of cells or particles. Currently, in recent years, she has studied connos in the Bushman land using an MSG grant. She has also done some genetic work with Andy Young, who many of you know, again, with onconophytums, and her latest MSG grant covers work done in the basis of today's talk. And today's talk is adaptations in Azoaceae to arid stress in Namaqualand. So I'd like you to welcome Robin, and I will ask her now if she would be prepared to share her screen.
Perfect. Thanks, Peter, for that introduction. Can you hear me? Is that all right?
You're very quiet at the moment, Robin.
Okay, I'll try to speak a bit louder. Is that better? Is it still quite quiet? I'm always having trouble. Hang on.
We'll have to turn our volumes up for a bit.
Okay, I'll just speak as loud as I can. Sorry.
That's perfect. Thank you.
With Zoom. Okay, I'm going to share my screen. If I can find it. Share. Okay, can you all see my screen now?
Yes, thank you.
Perfect. Okay, so good afternoon, everyone. As Peter kindly introduced me, I'm Dr. Robin Powell. I do want to just start this presentation off with a warning that I have, well, two kind of warnings. Firstly, I've recently given birth, so I am working on very few hours sleep, so if I'm a bit garbled in my speech, please forgive me. I am trying my best, but hopefully I'm able to kind of give you enough that you can understand what I'm trying to say. And then secondly, I am a slight imposter here in the sense of I'm very much, do a lot of science. I'm a terrible grower. Yeah, I seem to not be able to grow much. It's really quite embarrassing, but fortunately I work with a lot of people that are very happy to grow a lot of the mesems for me, so that allows me to continue with my research. I do try, and I am getting slightly better, but in my small London flat there's also not a lot of space for me to be able to do all these bits and pieces and, you know, have all the good things allowing me to grow these mesems. Okay, so as Peter mentioned, I'm based at Kew at the moment, but a lot of this research and specifically the project I'm going to be talking about was through collaborations with Liverpool John Moores University, which is through Andy Young, and also Mesemb, which is Kirstenbosch in South Africa. So Peter gave a really nice background to me. I mean, yeah, it's quite, I know it sounds quite technical, but I thought I'd just anyway give you an introduction into how I moved into mesems and a bit of my background and the work I do now and how I've kind of led to this point. So I was born in South Africa, grew up in South Africa, and I was very fortunate as a young person. My family traveled a lot around South Africa and often to very remote places. So my dad would often work with the mines, and we'd go to these very obscure places in the middle of nowhere. But this kind of intrinsically made me very interested in nature. Of course, when you're a young, you know, especially teenager, out in the middle of nowhere with nothing to do, you start looking around, start walking around, and I really developed a passion for nature and the outdoors. So when I got to university, I decided to do my undergraduate degree in environmental science. So I was quite interested in many different things around nature, which then moved me into doing my master's in botany at the University of Cape Town. This project was focused more on the fynbos, and I was looking at long-term changes, but it was very interesting. From there, I then was placed for an internship at Kirstenbosch for 1 year. And this is really where I came into contact with mesems and Azoaceae in a very kind of full way. My internship was based on systematics, so taxonomy and phylogenetics, the DNA analyses of mesems. And the reason I was given this project is because the family Azoaceae is actually identified as a top priority for taxonomic research in South Africa, because we have very little concrete published taxonomic evidence in the literature sometimes. It's a bit mixed, of course, but in terms of herbarium specimens and things like that, there's a lot of old stuff, but not necessarily as much of the new stuff. And that's also how I linked to getting linked with the MSG and Stephen Hammer and Andy, was because I know a lot of the growers, as you all are, have an amazing amount of resource and knowledge on these plants. And unfortunately, a lot of the herbaria are lacking this information. So really working together with people like Chris and Andy and Stephen have really helped me in my research, and I wouldn't have been able to do a lot of it without them. So coming back to that, after my internship, I then started my PhD. This was focused on a group, a clade, so a genetic clade of closely related genera. The main one was Conophytum. In that group, I did a lot of research on Conophytum, but also, as Peter mentioned, my publications around Caryopsis, Elenfeltia, Odontophorus, as well as Arenifera, Octopoma, and Schlecteranthus, which are the shrubby genera. They're not as interesting as the Caryopsis, I suppose. Not many people look at them as much. And then in that group, there was also the clades, the monotypic genera, Inagante, Namaquanthus, Rishianthus, and Gentinobotria, which I saw Matt showed really nice photos of earlier. So I looked at, in these genera, I looked at the relationships between them using genetics, but also understanding the morphological characters. So, leaf shapes, flowers, capsule characters as such. I also did a bit of anatomy. And one of my big things that I think that I really enjoyed was also using the ecology of the plants and how they're growing together to have a better understanding of the evolution of the group and how to tell them apart. But then moving on from my PhD, I then, as I did some genetic studies during my PhD, I really wanted to get deeper into the genetics. And so that's when I had a postdoc with Andy at Liverpool John Moores University. And that was 1 year. And really there, what we looked at is genome size. Genome size seems like a very abstract concept, but I'll get into that shortly. Don't worry about it too much. But basically, it's just the amount of DNA. The reason we wanted to look at this is, as diverse as Azoaceae is only one measurement recorded on a database for genome size for Azoaceae, and that's in Mesemb crystallinum. So really, we wanted to see what the variation in genome size was in this very varied plants family. And then, so that was about a year. And following that, how it always works in science, you think you're going to answer one question, and you come out having a lot of other questions. So moving from that, I then was fortunate enough to get a position at Kew doing research specifically in genome size, and now I work on all different types of plants and fungi. So I work and collaborate with people across Kew. But of course, I have a very vested interest in Azoaceae and succulents in general. So Crushulaceae specifically, but I also would like to really move into Cactaceae as well. And my kind of pet project is looking at genome size within these three families and really trying to understand it a bit more. So that's where I am now. So I've been talking a lot about genome size and mentioning it just to give you, I know I've got quite a little bit of terminology here. I know this is quite a wide audience with all different skills and backgrounds. I'll try explain things as best possible and as simple as possible, but please don't feel excluded or left out. And I'd be happy to kind of give any clarification if people need it. And the reason I am bringing this along is because it links quite importantly with the project and why I'm doing this project. So yeah, hopefully I can explain it in a way that everyone understands. And Yes. So genome size is actually the total amount of DNA in the nucleus of an organism. So as you know, all living organisms have DNA, and this DNA is housed in the nucleus. Don't really have to worry about that much. But what is really interesting, and I hadn't even really thought about this until I started this work, is that genome size actually varies across, so the amount of DNA within the different organisms varies across the range. It's not just a set amount of DNA in that sense. So for instance, humans, if you look on the left-hand side here, if you look on the left-hand side here, humans actually have quite small genomes. There's no correlation between organism complexity and genome size. So it's not the more complex organism, the larger the genome size, or the other way around. It's really quite random, and that's really what we're trying to figure out. So here we can see the human has a really relatively small genome size compared to flowering plants, which has the widest range in genome size. It varies 2,400-fold across all flowering plants, which is really quite amazing, considering most animals just fall in this small range where humans are. And then this is really kind of our basis of a lot of the research at Kew. The record holder at the moment for the largest genome is Paris japonica. I just want to check, so if people are raising their hand, is that towards the end, or should I do anything now? Okay.
I think it's probably better if you keep going for the moment, otherwise you could be interrupted a lot.
Yeah, okay, that's great. Sorry, I just wanted to confirm. Okay, great. So the record holder for genome size at the moment is Paris japonica, which is just a kind of herb. So it's really interesting, and you can see Fritillaria, some of you might know it here from the UK, is also quite large. This is Fritillaria here. So it's really amazing, these kind of inconspicuous plants that seem to have massive genomes. So really what we're trying to do at Kew is look, why is this range in genome size, and what could be driving all these different sizes in the amount of DNA? Just briefly, because genome size seems like such an abstract concept, I just thought I'd quickly show you how we measure it, because it's quite simple. So we have the target plant, which is basically the species you'd like to know the genome size for. The reference standard, which we often use crops for, so it could be, we often use pea or parsley. We know the genome size of that plant because it's been sequenced. And then what we do is we just chop it all up, and that releases the DNA in the nuclei. We then filter it just to get the large bits of leaf that we don't care about anymore. And then we add a stain, which stains the nucleus, which obviously contains the DNA, and that runs through a flow cytometer, which is this machine. Basically, it's just a very complex vacuum with a laser, and as the particles run through the pipe, the laser then looks at the amount of stain to correlate the size of the nucleus, which means how much DNA. So a larger nucleus has more DNA. And then the software actually gives us this output, and we can see, for instance, if this is a species we're interested in, this is the one we know the genome size, and we do a calculation just to get that genome size. So again, I will promise move on to Mesemb, but just as a bit of background, genome size is influenced by a lot of factors. We're not 100% sure. A lot of this is speculation because it is quite a complex, messy thing. You know, it's on a very microscopic level. We can't really see what's going on, but we have kind of some understanding of the ecology and how genome size then plays a role in that. So genome size varies across different altitudes. Very important for me and my research is that temperature has a strong influence on genome size, specifically in arid, hot areas. Genome size tends to be a lot smaller. We're not 100% sure why this is so, but that seems to be the trend. Life cycle as well. So long-lived plants such as trees and such can have much larger genomes because they have a lot more DNA. So every time you have to copy that DNA to create a new cell and to grow, if you have you need a lot of time. Whereas if you, for instance, an annual or grass, say, you don't want to have a very large genome because you have to grow quickly. Having to copy that DNA every time to grow another cell is going to take a lot of effort and a lot of time, and that just kind of restricts it. Nutrients are also important because things like nitrogen and phosphate are used to build DNA. So if you have a lot of that available in the soil, you don't really have limitations in terms of how big your genome can be. So high nutrient soils may allow for really large genomes, which is what we actually see in Fritillaria. Okay, I promise this is the final kind of really sciencey, extreme terminology slide. I know this is, like I said, I do apologize for all this terminology, but it is important to understand why this project was so interesting. So I hope a lot of you are familiar with DNA replication. So we have in a cell, usually we have chromosomes, and these chromosomes are then copied, so duplicated. And this is obviously when the plant is growing or creating a new cell. We then have it gets duplicated, and then it divides, and then we have two cells. So this happens all the time in every kind of living organism. So this is the general cell cycle, and most humans and animals are diploids, so we have two sets of chromosomes. But really interesting in plants, they often are polyploid, so they have multiple sets of chromosomes. Here you can see we have, 3 sets instead of just 2, or just 1 set here. And polyploids are quite important. I think a lot of you might be quite familiar with them. Polyploids are often crops. So basically what's happened is over the many, many years, people have selected larger fruits or larger flowers, which are generally the polyploids. So for instance, in strawberries, if you have the wild strawberry, you can see the fruit's very small, whereas the strawberry we eat is really big. And that's been selecting for polyploidy because the larger fruit always tends to be a polyploid. So the strawberry we eat in cultivation actually has 6 copies of the wild straw, 6 times the amount of copies of the wild strawberry. And then the final thing, and this is really what I work on, is endopolyploidy. So it's not just having multiple sets of chromosomes, but what happens is the DNA, when it duplicates, it makes the multiple copies, but instead of splitting into cells, it kind of gets stuck into the cycle and it never actually splits. So it just keeps adding and adding and adding and adding multiple cells. And if you can imagine at this point where you've got multiple copies, every time you keep adding more copies, your cell's just getting bigger and bigger and bigger. And so we think endopolyploidy is a way for a plant to make a bigger cell. And I'll explain why that's important in a bit. So as I said, polyploids, polyploids can make flowers bigger and fruits bigger. And endopolyploidy specifically in succulents are really important because, as we know, a lot of them have these water storage tissues below the epidermis. And what we think, although we haven't really proved it yet, but we think it's quite a good link, is in succulents such as Azoaceae and Crasulaceae, you get large amounts of polyploidy because they're trying to make these really large water cells. And so if you have a larger cell, you're able to store more water and you become a lot more efficient. At the moment, Azoaceae, Mesemb crystallinum is the record holder for the most endopolyploidy recorded in any plants, or actually anything, because endopolyploidy doesn't really occur in insects or animals. And so that's really why I looked at endopolyploidy in a lot more species of Azoaceae as well as Crasulaceae. Yeah. So as you can imagine, as I work on Azoaceae and Crasulaceae, and I know we have had some discussion around the succulent Karoo biome, but a lot of, well, actually all my work is centered around the succulent Karoo biome. It's one of the 25 global biodiversity hotspots. It's an arid winter rainfall region of South Africa. It's a mega diverse center for succulent plants. And Azoaceae really dominates this region, and often the species are found together with Crasulaceae. Another reason that I really enjoy working in the succulent crew, not just because the plants are there, but it's because they have a lot of different habitats, which makes it even more interesting. So for instance, you get, these kind of flats of quartz flats, and then quartz ridges, different species occurring in different parts of the landscape. You get coastal species, you get species, okay, this is a bad example of Lipoltia that's really kind of died, but you get species that are really occurring at the edge of existence in many ways. And then you also get species that are thriving in relatively higher altitude, fog-covered, kind of wet habitats. So it's really this contrast of different habitats, different species occurring in these habitats, and that kind of thing. And then, of course, as many of you know, the stone plants are really iconic group of the Azoaceae. They themselves occur in these very interesting quartz habitats, which are quite unique and make them really interesting. Even in the fact that they're miniature, there's a lot of interesting adaptations around them. So it's a very logical way to get interested in them. And then, of course, there's other habitats, such as where the Sphenistroria grows, very sandy kind of habitat, and then Conophytum marginatum growing again in quartz, but rather quartz blocks on these ridges. And another reason why Azoaceae and Crasulaceae are interesting to study specifically in this region is that you not only have different habitats and diverse species, but you also have different what we call functional groups. So you have miniature kind of conophytums growing alongside shrubs, and understanding how these different species and different groups are able to coexist and not overlap with each other, but also potentially, do they have different mechanisms of survival? Do they all survive the same? How do they take water from the landscape? How do they survive with drought? That kind of thing is really interesting to me. And I haven't got into that much detail with them yet, but I'm really excited to kind of move further along with that. And so coming on to the project itself, it really focuses on Azoaceae and Crasulaceae, specifically looking at genome size and endopolyploidy. As many of you, I am sure, are quite aware, but just to point out that the reason I chose these two families is because they really, really grow at exactly the same, in exactly the same habitat alongside each other. So here we have Conophytum calculus growing alongside Crasula deceptor. And what's really interesting around this is what my real question was are these species of these two different families, are they doing the same thing to survive in this really dry, arid landscape, or do they have different mechanisms of survival? What's the kind of, how they're able to survive together and not outcompete each other? That kind of thing is really what interests me. And again, here's an example of a Crasula here and a Monolaria growing together. So it really gives us such a nice model to try and understand, with exactly the same conditions, how are things different or the same? So, no, sorry, let me just move this. So assessing genome size and endopolyploidy in Azoaceae and Crasulaceae. To do this, I did a sampling field trip in 2019 that was funded by the MSG. I went out with 3 students from Kirstenbosch. Christian on the left here is currently working on Chrysanthemum. He's doing his master's on it. He's working on a smaller group within them. I think he's close to completion, although now with lockdown, I'm not exactly sure how much progress he's been able to make. We're hoping he also moves on to his PhD to complete Chrysanthemum and get really nice taxonomic keys, etc. for it, which hopefully will sort the family out, the genus out slightly. Shakira in the middle is also completing her master's, but focusing on a group of antimimers. Again, quite a tricky group, if those of you that know them. And then Liada, who's next to me, had just started his PhD working on the whole genus of antimimer as well as relatives. I am not actually sure how far he's made it so far with COVID lockdown and such, but I hope he's also going to make really exciting kind of progress on that group. And then being at Kew, I was also very fortunate to be allowed to sample the living collections for my research, and it was very, very convenient having the collections right there next to the lab. So I'd often go to Paul Rees and go collect a plant and then measure it basically straight away in the lab. So that was great. So following all the fieldwork samples as well as the greenhouse collection samples, I had approximately 350 samples of azoacea and crassulacea measured. This, I do want to say, I did not do it myself. Firstly, a lot of this was done by Andy when I was together with him in Liverpool, as well as I had a visiting researcher from Iran who came to help me. And the main part of this project analysis was done by my MSc student, Kate McRobbie. So unfortunately, just as she started her project, lockdown happened, so we had to adjust her project slightly, but fortunately we had all these samples that she could work with. So the first kind of comparison we did with these samples was to look at a general comparison of genome size and endopolyploidy in azoacea and crassulacea. And then secondly, what we wanted to do was across these biota sites, which are these long-term monitoring sites where they monitored climate and soils and things like that, but also had a species list. We wanted to look at species that occurred at these different sites and see whether they had differences in genome size and endopolyploidy, specifically the Knashflucter kind of associated sites versus the ones that were more north. Because initially from my fieldwork, it seemed like there was some Knashflucter, there was something going on in the Knashflucter at least. And so what we found, or what Kate's data showed, analysis showed, is that this is genome size, these box plots on the left. Genome size seems to be, we can see the box is quite narrow. So genome size doesn't range a lot in azoacea, it's quite restricted. It seems to be very consistent, nothing really too exciting in that sense going on. Whereas crassulacea, there's a lot more variation, which is quite interesting. There might be more polyploidy in crassulacea. We're not sure. We need to do chromosome counts to get that information. But there's definitely a difference between genome size in azoacea and crassulacea. Yet when we look at endopolyploidy on the right, we can actually see that despite the fact that there's differences in genome size, endopolyploidy seems to be quite similar between the two families. And this maybe suggests that because they occupy in the very similar habitats, they're under the same kind of stresses. Oh, what I didn't mention is endopolyploidy is often it makes bigger cells, but it's often a link to stress in an environment. So things like hereditary stress, of course, trigger it, light stress, UV stress, high salinity. So all these kind of things somehow trigger endopolyploidy, and plants are able to use it as an adaptation to deal with these stresses. So the fact that the two, that azoacea and crassulacea endopolyploidy is quite similar, maybe suggests that because they're under the same conditions, they're using endopolyploidy in the same way to kind of, what's the word, to cope with the stress of the environment. And then this is just the findings from the site comparisons. So the species occurring in the NKV, which is the non-Knashflucter sites, versus species occurring in the Knashflucter sites. Genome size in crassulacea in both are larger, where azoacea is less. But what is interesting is in the azoacea in the Knashflucter, there is a little bit more range in genome size. We're not sure why that would be, potentially it could just be the species that occur there. Agarodoma does have a strangely large genome size, but we're not exactly sure why. It might again be a polyploid that we missed, or needs to investigate further. But yeah, it's just showing there's definitely differences there in genome size. But even more interestingly is when we look at endopolyploidy at these sites. Endopolyploidy in azo-AC and the non-clash factor sites is very variable. And I think that kind of links to the fact that if you think of the clash factor as a very kind of set habitat, whereas comparing at the other sites is very variable habitats, things on gneiss, not always quartz, higher altitudes, more wet, drier, those kind of things. So I think it makes sense that there's more variation in endopolyploidy, yet Crashulaceae doesn't seem to have that same kind of variation. And then the Knash factor sites, the azo-AC actually shows higher levels of endopolyploidy. This is again really interesting because maybe could that be linked to the high salinity of the soils? Is it linked to potentially the high UV? So when the sun shines on those quartz stones, does the reflectance increase the UV, creating a kind of triggering this endopolyploidy? Again, that's not really seen in Crashulaceae, so it's quite interesting. A lot of this is like preliminary findings because we could only measure that many samples before lockdown. So there's a lot more research that we need to do on this, but it is showing that there's definitely some differences, which is, I think, quite interesting. So in conclusion, despite the fact that azo-AC and Crashulaceae occur in the same habitats, they show different adaptations to stress in the environment, really. So I think the fact that genome size varies differently in azo-AC and Crashulaceae, endopolyploidy is kind of similar, but not always the same, I think suggests that these two families have different evolutionary histories, so and colonization. So potentially has Crashulaceae, we know Crashulaceae is slightly older than azo-AC, although in that region not really that much older. Is Crashulaceae kind of using polyploidy or genome size rather as a way to kind of colonize habitats? And then perhaps azo-AC is using endopolyploidy more, or things like that. It's just, we're still trying to unpick it, but it does definitely seem like, although they're occurring in exactly the same habitat, they're not doing exactly the same thing. And then this actually might have implications for conservation of the species. So different families might need different strategies, especially under future climates. So if, for instance, azo-AC is able to use endopolyploidy a lot more efficiently, and things get warmer, does that mean azo-AC is going to survive better than Crashulaceae? We're not really sure, but I think that's something that needs to be thought about, especially when looking at conservation of these species. So enough about the very heavy science now. I'm sure there's a lot more. I'm going to be sharing a little bit more of what I found in just general pictures. I know that's what everyone enjoys a lot more. While I was out in the field last year, I discovered a new species of Dicrocolon. At least, yeah, it definitely is a new species. I'm in the process of describing it with Adam Harrower at Kirstenbosch, who'd also, interestingly, found it at a similar site. Well, not similar, like another site. And so when I showed him this plant, he said, oh yeah, I think I collected it as well. And he'd also thought it looked quite strange. So yeah, we're looking forward to describing it. Of course, my maternity leave, I'm going to try fit into maternity leave, but perhaps not. And then I'll also be doing that with Paul Rees at Kew, who's growing the plants for me, because as I said, I'm not the most successful grower. So fortunately, I do have herbarium specimens and all the important characters are there. So yeah, but it's obviously always, it's very attractive plant, as you can see. So hopefully it will be quite an exciting addition. And then I just thought I'd just share some general fieldwork photos with you. I think, you know, I always enjoy being out in the field. I do miss it, especially this year and especially today when it's so grey and rainy here. This would be, well, a bit later, but I would have typically already been in the field this year. Yeah, so this is just Namaqualand on the way to Subatsfontein. This was last year on the road from Alexander Bay to Extienfontein was really, really, really so dry and really sparse. It was quite sad being out there. Like I showed earlier, that Leipolter that had just basically died. I don't think many of those populations are coming back. I did speak to Peter about Peter van Wyk. Those of you who know him, he seemed quite upset about all of this. However, it seems from what I've seen on his Facebook and things like that, that they've had really good rain this year, and it seems to be a really good recovery. So unfortunately, we're all stuck here and aren't able to go see the nice blooms and things, especially in the Richtersveld. I think it's so special. But yeah, hopefully a lot of the populations will recover and new seed would set and hopefully be able to establish themselves again. But let's see. Springbok, those of you who've been to Springbok, it's just such a nice place. I think it would be my second home if I could be there more often. You know, even just in the backyard of the place you're staying, you end up seeing so many nice plants, always collecting. You never know what you're going to find. Yeah, so that's always exciting. And of course, you get the most beautiful sunsets out there. This was on a hill at Smoornskadu. Bushmanland is just a really special place. Smoornskadu itself is an interesting farm because it lies on the border of this winter-summer rainfall region. And a lot of the times, although that sounds like they're going to benefit from both winter and summer rainfall, actually most of the time they get neither because they're really just on that boundary. So yeah, they hard-working farmers out there. They really struggle with the drought there, but you have the most interesting plants there. And the farmers are really sweet and friendly. Again, just fieldwork, road verges, often very exciting place to see plants. This was last year with the students, and Christian, who was working on Chrysanthemum, was always in the road verges because the chrysanthemum just love growing in these very disturbed places. So that was really fortunate for him. He didn't have to do much hiking. Of course, Liada and Shakira and I that were looking for more diverse and interesting plants, and especially the antimimas, I had to do a little bit more climbing, although it's not so difficult in that region. And while we were out in the field, we were treated to some rain, which is really unusual, but also really beautiful blue skies, which was always lovely. And then the last day of field work was a lot of fun. We're always putting together all our collections, have the obligatory wine, enjoy a little celebration. And we were fortunate enough to see a rainbow, which was very special. And then finally, this wasn't last year's field trip, but a few of my other field trips, I just thought I'd share some memories. I'm always fortunate to stay in the nicest places with such lovely people. It's really such a special place, and I hope a lot of you get a chance to go there. I know things, especially now with COVID, is not the greatest, and I'm sure a lot of people are struggling there. But hopefully things will ease off soon and we'll be able to visit again and enjoy the sunsets and the beautiful scenery, and of course the lovely plants. And then closing off, this is one of my favorite places to stay in Namaqualand or Bushmanland. It's Achab. Those of you may know it. It was Wim Gerard van der Heever's place, right next to Gamsberg. Unfortunately, he sold it to the mine, as far as I know, and they are apparently going to be using it as an offset for all the development of Gamsberg. However, I'm not sure what's happened so far. I know Andy had some discussions around this or some consultations around this. I'm not sure actually where they got to on this point, but yeah, it was a really special place. I loved staying there. Very interesting plants again there, a lot of endemics, and just lovely people as always. So yeah, finish off with just a view from one of my favorite places. Thank you.
Thank you very much indeed, Robin. You've made an absolutely superb job of trying to make what is very scientific work understandable to the majority of us who haven't got a clue. I'm a lot wiser than I was before, and I thank you for putting that over to us. Thank you very much indeed. I will hand back to Ivan now.
Are we going to take some questions first? I think we've got a couple of minutes. We can take some questions first. If people know how to raise their hand, if you open the participants bar, if you run your cursor towards the bottom of the screen and open the participants bar and then raise your hand, there's an option in there where the three towards the bottom of the participants bar to do that. I don't know if John's able to see them. I'm not at the moment, if anybody's raised their hand.
Roland Debenham has raised his hand.
Roland, would you like to unmute yourself and ask a question then? You're muted, Roland, still.
Unmute yourself, Roland.
Yeah, I have. It's all right. I need 3 clicks of the mouse to do that. Question for Robin. In a polyploid, are all of the DNA strings identical, or are there mutated single polymorphisms in some of the strings?
So I'll admit I'm not an expert on polyploidy, but as far as I understand, it's normally identical duplicates of the same chromosome set. But that's not to say, although it seems always like a neat, compact, nice package of a chromosome, I think there often are mutations, but it wouldn't necessarily be a massive mutation in the sense of how we would know as a mutation. You'd just have a few bases that could be changed. But
sure.
Like I'm not a polyploid expert, so I'm not sure. I don't always work on the chromosome level. Yeah.
Can I have a follow-up then? Have you published anything yet which we can access either directly from you or on the web?
So I published with Andy our AZOAC paper on genome size and endopolyploidy and conophytum. I think I can share that with you. I'm not 100% sure. Otherwise, if you go through ResearchGate, you might be able to connect to it.
That's in the Botanical Journal of the Linnaean Society, yes?
Yeah, exactly.
Yes, that paper.
Yeah. So that's
I might be able to get access through the Bodleian Library. Thank you.
Yeah. Okay, perfect.
Okay, anybody else with any questions? If somebody has, would they please unmute themselves? I can't see anybody else's hand raised, but if anybody else has, please do unmute yourself and ask. We seem to have lost Matt, so if anybody particular wanted to ask him anything.
Matt's here, I can see Matt.
Oh, you're there. Oh, I'd lost you. Sorry. Matt is with us. So if anybody wants to ask Matt any questions from his presentation as well. Then...
David Lambie had a question and he's unmuted now. Maybe he could go.
I had a message that there were 2 raised hands, but I had no names.
Yeah, so I did raise my hand. So first of all, thank you very much. That was brilliant. I really enjoyed it. My question is a really stupid one, or at least I think the answer seems so obvious. I'm surprised I'm asking it. But why does a big genome necessarily mean a big cell?
So if you remember the slide with the multiple copy, basically, if you're making more copies, you just need more space for that DNA. And because the cell is a little bit more plastic, it will then just create more space. You know, if you can imagine it just forcing the cell to be bigger. So that's basically why it makes a bigger cell, because you just have multiple copies and things taking up that space. So you just need a bigger kind of area, if that answers.
But that's presumably in the nucleus.
So, yeah, exactly. So as the nucleus gets bigger, then everything proportionally, it's slightly tricky with a vacuole. You don't always necessarily have a larger vacuole as well, and it could become smaller, but generally the rule is if a bigger nucleus, a bigger cell.
Is that because there's more kind of action with the sort of the protein generation, because you've got more bits to create it? So it's just more lines in the factory to create food and...
Yeah.
Okay, great, thank you.
I've got a question here from Nick Evans. He can't see how to raise his hand. A question for Robin. Is genome size solely related to the degree of polyploidy, or is chromosome size also a factor?
Yeah, so genome size, polyploidy and genome size are often linked. Obviously, if you have a polyploid, you have more DNA, which makes a bigger genome size. Chromosome size, let me think about that. Yeah, I think it is sometimes linked, but you also have another part of the genome, which isn't just polyploids, but it's called transposable elements. And these are just kind of leftover bits of the DNA that it's kind of like messy bits, but they also add to genome size. And so that's why in polyploids, you might have, say, let's give you a random number. So in the diploid, non-polyploid variation, it might be 100 base pairs. And in a polyploid, you'd expect 200, but sometimes you have 240 or say. So it's quite complex to unpick all these bits and pieces, and it's quite heavy genomics, which I would admit I'm not an expert in. I do a lot more of the techniques around it. You know, it's heavy epigenetics, but yeah, there's a lot of things that contribute to it. Basically, in a very neat, compact form, you could say, okay, polyploidy, higher polyploidy means bigger genomes, or bigger chromosomes potentially could mean bigger genomes, but there's a lot more kind of messy factors around that.
I'm picking up questions in the chat bar. I've got one here from Frankie Richardson, which could be addressed, I think, to all the speakers. The question is, have you got any books or resources for studying AZO-ACA?
I don't know if anyone else wants to answer that. Basically, I use Heidi Hartman's AZO-ACA A to Z. That is like my kind of Mesemb Bible, but I also use the Mesembs of the World as quite a nice reference for genera. Yeah, that's basically what I use, and any other literature I can get my hands on. I don't know about everyone else.
Any other of the speakers got anything to add? Right, in that case then, I've got another question. Again, it's aimed at Robin from Michael Gatekoski. I hope I've pronounced that right. Could polyploidy simply be a way to duplicate, triplicate, and so on copies of highly expressed genes?
Yeah, it definitely could. Like I said, I'm not 100% expert on polyploidy, but yeah, it's just you'd want to. So in a polyploid, you are going to be duplicating the whole chromosome, so you're not going to be just duplicating selective genes. But obviously, if you want, I don't know, a gene that expresses, for instance, a red color or so, I think a plant could be selected to duplicate that gene specifically as well.
Just going back to that last question, sorry, about the books. Matt put up quite a new book. I don't know if you want to just say a few words about that.
Oh, I put up Secrets of Namaqualand Succulents, which is a new one by a guy, Florent Grenier, who's been very active in the past few years doing field work in South Africa. And it's, you know, quite a nice book. Kind of it is idiosyncratic, but it's a fun book.
Thank you.
I just caught somebody and their name was just disappearing. Andrew Butchers, I think it was, had raised a hand. If Andrew would like to unmute himself and ask his question.
Hello, thanks. It's probably a complex question. Many years ago, I studied A-level biology and geology. That was back in the 80s. I'm just wondering, looking back at it then, it seemed that the theory of evolution was, you know, quite well documented and a lot of evidence for it. Why hasn't it been declared a fundamental law of biology by now?
Are you asking me that question?
Either you or Matt, or you and Matt.
Yeah, maybe Matt could answer that better. I mean, I think it's well accepted now, but I'm not sure. I mean, I really work on plant genetics, and a lot of people do that. I don't know if Matt has any insights.
Okay, thanks.
Sure. I think there's a difference between people, the way that people use the term theory and sort of common language and how it's used in science. In science, a theory is kind of a really broad set of facts and the explanations that sort of tie all those facts together and enable predictions and hypothesis testing and all those things. So, you know, like we have a theory of gravitation, which is, you know, Einstein's theory of general relativity. And it's a theory because it explains gravitation in a certain way with warping of space-time and all that. It doesn't mean that gravity is in doubt. It's so theory in science is quite a different sort of thing maybe than it is in common parlance, and I think that may be leading to some confusion there. Does that answer your question?
Sort of, thank you.
Right, well, if there are no further obvious questions, can I ask that we move on to the next stage then? Brian, are you ready to give your short presentation? I know we've got a little bit out of sequence here, and we've not done proper thanking of our speakers at this stage. But if we can do the presentations and the thanks at the end, I think that might be the best way around it. So, Brian.
Okay, thank you very much.
Brian's another long-standing member of the MSG and lives in Glasgow. So introduces yet another country into our speaker list today. And he has the opposite type of climate to work in that it's a lot colder, wetter, and not as much light as the rest of us get in England. Brian, thank you.
Okay, thanks. So a big hello from rainy Glasgow. I'm just going to quickly put up a couple of pictures of Banstead and the Northwest Mesemb show. That's what we're missing this time of year. So it's a great shame not to see all the people face to face. But looking on a positive, it's nice to share our fabulous hobby with people further afield. So hello to everybody who I don't normally know. But tonight, I generally grow an awful lot of Mesembs, just a mixed collection. And it can be quite difficult, as Ivor alluded to, in Glasgow, because we are known for our dull weather and lack of winter light. But tonight, this talk, I'm going to talk about a little bit of observations of lithops leaves. It was just the last year or so, I've been starting to look at the dried leaves from my lithops, and it's inspired this presentation. So I'm going to start off basically by showing a few sheathing plants. All our Mesembs go through cycles of growing, and it's important to follow those cycles. Obviously, the most noticeable of cycles is those shown by the sheathing plants. So these are just some of the examples. So as we've seen before, Chirodopsis go through a dormancy period and then full active where they'll burst out of the sheaths. Conophytums are again one of the genera which uses a lot of sheathing to protect itself through the harshest of elements in habitat, and quite fascinating. Very much quite ephemeral, some of the sheaths on Conophytums, but amazing how they can sort of like emboss the epidermis into the skin, and that goes through almost like lizard skins. So it's always impressive. So get your magnifying glasses out and get into the greenhouse and have a good look at that. So you'll see there, this is on to lithops now. So this is Lithops colorum doing test book sort of like transitioning, going from a dormant period, recycling and changing through. But it's very difficult for me to achieve this in Scotland because obviously the climate. Those of you who are in very warm conditions, sunny conditions, can probably let your lithops dry out. I'm lucky if I get this, jump round about the beginning of June, July. The old leaves will become leathery and the new ones will actually emerge beforehand. So this is fair enough. That's the way I have to work with it. And usually with it by the end of June or July, they will have dried out completely and they'll be ready to think about flowering. So I've got, obviously most of you will have seen a cross section through a lithops. This is one I shot on film a long time ago. But just to basically show what we've got the hard, tough skin on the outside of the lithops. The next layer in is the active layer, which holds the chlorophyll. Obviously, the top part of the body is clear to allow the light to go through to the lower surfaces. Now, this area here is quite interesting. That's the water storage area. And you can actually see to the top left, we've got some bubbles in there. I'm wondering if these are actually internal bladder cells, storage for water. And we can see what looks like veins running through. So basically, we've got two modified leaves which are fused together. The central point is, which goes from the fissure at the top of the body, is just an airspace. So obviously, when the plant is absorbing more water, that space will diminish. As it's becoming dehydrated, the airspace will probably become a bit bigger and more apparent. So that's the cross section. Basically, like I said, we've got two leaves. So I thought to show the enormity of what's happening, I've just gone into the garden and picked out two. We've got two leaves, and basically they've folded up on themselves, and the tips have gone over. So what we're actually seeing when we see the lithops is the underside of what would have been traditionally two leaves. Fused together along the margins, and then with a whole load of water storage material inside. Quite fascinating, and a pinnacle of evolution, if you can imagine. Probably easy to understand when we saw the skelettium that we saw in Matt's talk, to see that. But from a garden perennial, it would be a big leap of evolution to imagine this. So looking at the surface of the lithops, that surface structure is very, very thick tissue. Fascinating at close-up. You can actually see the cells on the surface. That's the only area which is going to be exposed normally to the daylight. So it's structured to resist the evaporation and everything, but it still transmits light. So you can see this from light from below. You can see the light. It changes the perspective when you see a lithops lit from below. You can't really identify them readily. But if you turn it over, we can see the same structure we saw earlier, but this time in cross section. So we're seeing lots of veins that have been severed through that water storage material. We've got the chlorophyll area and that green ring there, and we've got the very thick rind, if you like, of the skin. Again, bisecting these tips, it becomes much more apparent the vein structure again. And these bubbles that we saw earlier, possibly bladder cells, I'd like to know if anybody's got an opinion on these. But on the left-hand side, we can actually see these bubbles that have actually cut through. It gives it an impression of a Swiss cheese more than anything. So my theory is that obviously this is a water storage area where the plant can actually quickly store water to avoid actual cell destruction if it imbibes too much, or it can utilize it very quickly, because obviously there's a lot of veins running through this storage material as well. Looking at the lithops there with many years of leaves, it's quite interesting to see the color combinations. The last year's leaves are sort of golden, the year before bleached white, and the ones previous to that are gone black because of probably sooty mold and things like that. I certainly wouldn't recommend leaving the leaves on in the UK, because what I have noticed is that they're capable of absorbing moisture from the atmosphere. So obviously, if you imagine if you live somewhere like Glasgow, where it's very dull for weeks on end and quite a high humidity in the air, these skins will actually absorb plenty of water and could lead to rotting in the winter, in the dark months, when we don't really want them to be too wet. Obviously, as well, when they're dry, it could be there could be lots of beasts lurking in there, pests, so potential pests. So, really, ideally, would remove those. The fact that they absorb moisture perhaps might be an advantage in habitat, because what I'm thinking is if with rain events or fog events, these leaves, old leaves, might absorb moisture, and with a slow evaporation later on, could create a slight cooling effect, perhaps, for the lithops, and make it a little bit cooler environment, more pleasant in such a hostile environment. So they could serve as a potential advantage in the wild, but certainly not in cultivation. So this is a leaf which didn't quite lose its moisture from the surface. I severed this from the plant after the new leaves had appeared through. And it's quite interesting to see the structure of the leaf. But more interestingly, at the bottom, we can see as the moisture at the bottom of the leaf has been drawn out or reused, recycled for the new leaves, it's actually created sort of a laminate effect. All the veins running through the water storage tissue are actually coming together and being pressed against the back wall of the lithops. So really, it's quite interesting to see that there's very little substance to that material on the inside. The main part of the lithops leaf is the outer wall, which is very thick to prevent it losing water. So on the left, we can see 5 dried skins, the inside edges of 5 dried skins. And to me, it's interesting because we can see all those veins have actually been pressed firmly against the outside wall. So if you imagine all that tissue from the outside wall right to the middle has practically gone to nothing. So it just shows you how thin the cellular tissue is in that space. So I would imagine that any air contact to the lining of that inner space would be very easy to transmit gases and moisture at all possible. On the 2 on the right, the 2 skins on the right are the outside edges of the leaf. So you can see it's very stable structure. It's quite rigid, and there's no sign of any veining whatsoever. Obviously, the crinkly bits and what you can see at the top, that's the remains of the shoulder of the lithops, and it's on the window. So obviously very thick tissue to actually fold up like that and not go too thin. So this actual image that really got me thinking about that aspect of lithops culture. We see a similar thing when we look at frutescens. Obviously, perhaps the bilobums and frutescens have got the most similar of skins to the lithops. But what's fascinating when looking back at this is that you can see on the enlarged area, it's almost like a 2-layer effect. The outside skin is fairly thick and crispy. The inside skin is very, very fragile looking, almost gossamer-like. And it's almost like a laminate with the veins running through the middle. But we can see that the outside skin is much, much thinner than the lithops because all those veins are actually protruding through the outside skin. So again, it just shows you how thick that skin is on the lithops. Lithops babies are very easily to get under control. They're so small, the skins are so thin, they can dry out very easily. No problems in drying those out, as we can see here. But as they start to mature, certainly here in Scotland, the moisture is so heavy in the air, if you like, it would be difficult to actually dry them out properly. So this is a good situation for me normally in the greenhouse in June to see the discoloration of the old leaves and shrinking down, shriveling down, and the new leaves looking firm. So that's what I'm aiming for. It can create a problem once you start getting clumps of plants because not all the heads lose the moisture at the same time. And so you'll have some that have not changed at all, some that are starting to change, and some that have already changed and lost the leaves. The one thing I've found is critical is not to try and dry everything off so that they all catch up, because you would be struggling with the first ones to lose the leaves would suddenly suffer because of that, and you might lose heads. So I always aim for somewhere in the middle. Once I've got some of the middle ones to actually regenerate and recycle, then I'll start to give water. The ones that keep the leaves for an extra year, I just allow to happen. But I have developed a technique to reduce that effect. These there are Lithops alcampii in April. Now this is obviously April, it's the critical time not to water at this stage. You can see how fat and bloated the old leaves are. If I was to water at this stage, there's a good chance that both the new leaves and the old leaves would still take up that water. And so it would be practically impossible to split the skins afterwards. And in situations like that, when a lithops has been overwatered, it might start stacking. That is creating more and more leaves, which is definitely not what we want to happen. But it's easy enough to actually avert disaster by actually severing some of the edge of the fissure. You can get a small scalpel or something and just create a little slit. And the pressure of those outer leaves being so fat and turgid would actually create the split for you. You know, you only have to start the split going and then it starts to open up. And the good thing then is to just let the air get into, let the internal parts of that leaf start to get in touch with the air, because then they'll start evaporating much quicker. Things like Norini can be a problem. They'll never be able to dry out the leaves of Norini. These gaping mouthed leaves tend to always work in the process of the leaves coming out and retaining the old leaves for a short while at least anyway. And you can see this one's looking particularly dry because these top new ones with the new leaves are even wrinkled as well. So I've perhaps been a bit mean with the watering on this one. Just to highlight the remedial process of getting rid of stacked leaves. This was a plant I got actually at a Banstead event, which was 4 years ago. Unfortunately, I didn't take a picture when I first bought it, but the outer leaves were tight as a drum around the outside. This plant had been watered with a collection of cacti, so it had been getting water throughout the summer, and the water had nowhere to go. So what I did was, as I described, I started to split the outside edge. And then that's when I started taking the pictures, unfortunately. But gradually, every day, I would start peeling them back. I wouldn't take the leaves off all in one go, because you really want to harden off the new pair of leaves below. But that inner surface, which is so thin and flimsy, evaporates very quickly. And so the water starts to lose very quickly. And then the next summer, the plant had completely forgotten its bloated past and flowered for me. And then literally 12 months after purchase, you would never have known that it was a potential stacker. So just finishing off now, obviously lockdown gave us a lot of potential for thought. And one of those was this lovely sensation of slow motion bubbles from a watering bunch of seedlings. So sorry if I've rushed through that. 10 minutes is an awfully short time, which I've realized when I've tried to whittle this talk down to a manageable size. But I hope you enjoyed it, and hopefully it's maybe give you some ideas to things to look at and to talk about later on. So thank you very much for that.
Thank you very much, Brian. That was really good. Enjoyed that one. Thank you. Yeah, everybody's nodding and clapping. Thank you. Again, because of the time constraints, I think if we can go straight on to Terry's final talk, please. And then we can get Peter back to give his final comments. Thank you. Terry, I don't know if you've unmuted yourself.
Yeah, I think I should be unmuted.
You have now. Yes,
and you should be seeing the full screen presentation.
Yes.
Okay, fine. Well, all that I've done this evening is basically let you see what my greenhouse is more or less currently looking like. So what I did, I went out there a few days ago with my camera and just took a few pictures. And of course, these are a lot of the plants which, if this had been the real Mesemb event, would have been put in the car to take to Banstead for a table display. But I found that flowering started very early this year, but there's still quite a lot hanging on, as you can see there. One thing I'd like to point out is the back of my greenhouse there, which faces southwest, I get white shading painted on it around about April. And come September, I take it off in 2 goes. So I've just gone over it with a duster and removed it to create the striped effect. And now as soon as we get a dry day, whenever that might be, I'll finish taking it all off. But my connos always, the area where my connos are, always have a bit of shading each summer. Now, some of these nice floriferous plants on the top shelf are in sort of section minuscula. We've got the very small heads, and they really do smother themselves with flower. And that's one of them, Consolation Leucophiae. And that's one of the other ones from up there, Rubra lineatum. And it's interesting that both those stocks have been in cultivation for a very long time. But some of the exploration that Chris and Andy have done in recent years over the Table Mountain sandstones have really shown that both those species are a lot more variable than they might appear to be from the old stocks that we grow. And in fact, I sometimes have problems distinguishing Swanopolianum from Rubra lineatum when you sort of get things of intermediate size. I don't normally grow hybrids, but this is a little thing that Steve Hammer created, a Morniranum crossed with Terrigerum. And it was given to me, I think it was either Andy or Chris at the last Mesemb event. And it's a pretty little thing, I'll grant that. Now, another of Chris's past creations or christenings was a yellow-flowered Conophytum ratum, to which he gave the name Golden Glow. Now, over the years, you know, I used to sell connos until about 3 years ago. And over the years, I sowed quite a lot of seeds and raised them. And from all the sort of ratums which flowered for me, which were normally pink, I did end up with one yellow one you can see here. And I also did end up with some other, one or two other plants, a little bit like this one on the right, which are more sort of apricot in shade. So I put pollen from one of these apricot ones onto the yellow one. And that's a group of seedlings that you can see there. Now, excess seed of that did go into the MSG distribution. So, you know, others of you might have little groups of plants like this. And from these, some are pinkish, but there's 4 there which I've marked with, sorry, 5, which I've marked with blue labels now, which have got the pure yellow flowers. So what I've been doing this year is pollinating all this group of yellow flowered ones together to get seed which I hope should be predominantly yellow flowered. Few little puzzles and weirdos. A concentrating burgery here, which decided it was going to divide dichotomously, but didn't quite make it. So we've basically got a Siamese burgery, which is half joined and half not. And I'm fairly sure that when that emerges next year, that will be 2 separate heads. Over this side, we've got one of these many weird plants that come from the Reedkloof area. You look at it and you say, but is it an actipum? Or is it related to something like Sponsaliorum? And there seems a whole lot of weird and intermediate connos which grow in the Reedkloof area. So that's a little bit of a puzzle to me. I put this in because when I was selling plants, and even still, I've probably had more requests from people to obtain Conobasianum than almost any other species of conno. Now, the problem with this is that it is incredibly difficult to grow. I thought I'd conquered it at one point, and I had a 2-inch pot, 5-centimeter pot, with maybe 3 dozen heads in it. They got knocked back, and now I've just got a small amount growing again, which I hope I can keep. There was a bud there when I photographed it. The next time I checked it, there was a dead flower there, so it didn't actually last for very long. The only advice I can give to grow this is, 1, give it quite a lot of shade during the summer. And 2, most of these plants in the Rudii group benefit from frequent repotting. And when you repot them, pull apart whatever heads you can pull apart. Because in this group, individual heads have roots on. So it is possible to pull things apart, each with roots intact. So that's my only 2 suggestions to grow some of these very difficult things in that Rudii group. Now, this is the season when I try to pollinate plants. Having given up plant selling, I can spend more time in the autumn doing things like pollination. And my aim is to produce as much seed as possible for things like the Mesemb study group distribution and the BCSS distribution. Now, because I've got very limited space, the best I can do is take them out from their near neighbours, as you can see done here, and just put them in other parts of the greenhouse. These are pots of South African bulbs just emerging, a couple of Taylorianums here, a batch of cubicums there. And then I hand pollinate them using my infamous human hair technique. I think under my conditions, I get very little pollination, inadvertent pollination from flying insects. Where I do get inadvertent pollination is from Western flower thrips. And I hope that Western flower thrips doesn't travel too far. So by isolating them from anything that they're easily crossed with, I'm hoping that I can keep good seed, but I obviously can't give a 100% guarantee for the seed I produce. these three little Conophyta mirabile, which I bunged amongst some sales Haworthias, which are sitting there, which are going to be really big by the time I can sell them at any meetings again. Now, the night flowering ones, I don't want to go wandering out to the greenhouse at night. So I bring these up to the conservatory, which is just outside our living room. So, you know, this evening in between, say, Strictly Come Dancing and Britain's Got Talent, I can just pop out there and pollinate some of these night flowering plants. So here's a nice batch of Comptonii, which is the Papco's fontaine, which I find the easiest to grow. And here's some of my Obcodellum Ursprongianum, which are sitting there. I visited the farm Lochenberg about 25 years ago, and Mrs. Locke, whose family the name is named for, showed us these plants growing there, and I was allowed to bring back a capsule from which I have raised all of my stock, but I keep inbreeding it. And the ones with the darkest and most prominent markings, I breed together. And I've now got a couple up the back there, which are really getting towards some of the black minimums that you can get, the things that Chris calls blackout. And my stock of Obcodellums, I have got a load of seeds sitting around from last year, and they will get sent to the MSD seed distribution for the coming season. A little part of my shelving has got the Ophthalmophyllum group on it. These all tend to flower a little bit later, I think, than many of the other conus. But one little problem that I see, well, first of all, the Ophthalmophyllum group is very difficult to name. And I'm sure there are too many names there, really. But we're actually growing two things here, Conophytum pubescens, onto the same name, which are really rather different to each other. The form on the left grows west of Plattbacchies, this grows north of Plattbacchies. The original description calls for an elongated pubescent plant. Well, this one is elongated but smooth. This one is squat but pubescent. So, you know, would the real pubescent stand up, please? And I grew one or two other things. I grew a few lithops, but not very well. But go out 5 o'clock in the evening at this time of year, and there's nice flowers showing, and quite a nice scent comes up from some of these too. In my other metal greenhouse, I do keep some of the lithops cultivars. There's sort of Leslie Jamboree there, and there's some Olivaceae red olive there, and some the Pincot Zeniana. And I keep these for pollinating too. So I produce seeds of some of these cultivars, which, you know, they can go in the distributions and go for a few pence. But if you start buying them from Meta Gardener or somewhere, actually tend to cost a fortune these days. And just finish off with 2 slides showing you that I do grow other things. We've already seen this afternoon, but I grew a few Dinteranthus. This seed for the pink one actually came from Steve Hammer, and he called it Pinky, but I don't think that's a cultivar name which has ever been established. And something else in flower at the moment, which fits in with Catherine's presentation, I've got Gabeum Brownii in flower. And note that although I grew almost all my Mesembs in plastic pots, this one's in a clay pot. And if I give the slightest bit of too much water to Gabeum Brownii, it splits. So my answer to that is to use the clay pot. I split it sometimes, but usually, no, you know, I managed to get away with it. So that's my little presentation. And really, thank you for the 148 of you out of a maximum of, I think it was 199 at its maximum, that stayed the course. So thank you to all, and back to Ivor to finish.
Right. Well, thank you for that, Terry. It's been a good day, I think, anyway. I've had a very enjoyable afternoon. I'm sorry that we've not really been able to thank all our speakers to the degree at which they've managed to entertain and educate us. I have a big apology for that. Sorry it ended up being reordered, but we did get through everybody in the end. We only overran by 40 minutes, which wasn't too bad at the end. So thank you for John for helping to organise it, to all our speakers. And I'm going to pass over to Peter when he unmutes himself, please, for his closing remarks.
Thank you, Ivan. I'll be brief. It's been a splendid afternoon, and it's been a fitting replacement for the fact we lost our 2 Mesemb events. Sadly, we've not been able to provide any virtual plant sales, but there we are. There are many people to thank for this afternoon's presentation. First, Ivor, Suzanne and Terry for meeting and agreeing to put on the set of talks as a substitute for the happenings that should have been. To Ivor for all his hard work in engaging all the speakers with able assistance from Suzanne and Terry. Again, Ivor for making all the arrangements for the meeting, being able to be viewed by you and all via Zoom presentation. To John Schwartz from San Francisco for allowing us to use the Zoom license, which accommodated up to 500 people, and our 101 would have been no good. To all the speakers for giving their time and expertise in their chosen subjects to add to our knowledge and to provide admirable entertainment for us. To you, all you wonderful people, for coming to this gathering. No matter how much hard work is done by the planners in advance, it would be pointless if you did not come and participate. I'll just add what Terry said earlier. We are looking at the feasibility of providing a Zoom meeting for Mesemb enthusiasts every 3 months as an addition to our hobby. More about that in the weeks to come. If I've missed anybody inadvertently, I'm sorry, and I thank you that way. I wish you all an enjoyable continuation of your day or night or whatever it might be, but bid you a fond farewell. Thank you, Ivan.
Thank you. I shall leave this open for a little bit longer. We'll wait and see how people drift away. If people want to unmute themselves and to say anything further or generally chat or whatever, please feel free to do so. At this stage, the chat bar's still open and you can unmute yourselves and speak if you wish to. Thank you.
Brian, I'm going to throw one at you. I noticed in 1 or 2 of your skins the red dots. I thought you might have perhaps said something about those as well.
Were they in the lithops?
Yes.
Or the conophytum, obviously, there's a lot of tannins in the conophytum skins, which you'll see in a lot of the things like biolobums and things like that. Was it red dots in the lithops skin you were mentioning? Because
yes, that was the one that I noticed.
Well, I did see someone who was preparing it that like reddish spots on the leaf, which was didn't quite dry out, and it was really on the cut surfaces. So I was wondering if that was tannins that was leaching out from the lithops leaf. That's all I can think of from what you're saying. Obviously, the only other red parts on the lithops leaf were what was in the upper surface of the window of one of the shots I had. But I think that was just to do with the rubrications, if you like, of the islands and things like that. But obviously, there is tannins involved. It's like, I suppose it's like in autumn when the leaves fall off, the trees throw away all the rubbish, don't they? So the lithops will do the same with the skins as well. What they don't want, they'll discard in the leaf as well, in the dried skins.
Yeah, I found it very interesting seeing them there right in the middle of the tissue.
Well, that was the veins, and it was the veins that made me start looking at it a bit more. It was the dried skin, seeing the veins on the dried skin, but then putting them in association with the cross sections, seeing how many veins were running through the water storage area. I was not aware of that until I started looking at just the last year or so, which is quite interesting, I found. But it just also shows, I might have went too fast there, it just shows you how little substance there is to the tissue on the internal parts of the lithops, so that when you take the water out of it, there's hardly anything there. There's nothing. All those veins are getting pushed right out to the side. There's no tissue. So if you imagine the upper leaf surfaces, which are now on the inside of the lithops, they're very thin. The actual, perhaps only a cell thin. So there must be a lot of transfer of gases and water between water, through that surface, whereas the outside obviously is impermeable, probably.
Yeah, very true.
Well, it seems our numbers are falling away quite rapidly now. So if nobody's got any further suggestions, things that they want to urgently talk about, then I think we can ask John if he'd kindly close it down. Then he can go back to bed and get some sleep, having got up so early this morning to help us out with this. And the rest of us can go off and have a rest and something to eat for our evening meals. So
time to order your Indian takeaway.
However
good.
Yeah, however you want to eat. So thanks everyone for coming along, and as you say, watch this space. Hopefully through the TSG we may have something else to offer before too much longer. Through the MSG, sorry. Thank you.
Thanks from me to everyone who helped.
I think there are some interesting comments in the chat bar, quite a lot of them.
Yeah.